เป็นนกนี่ก็โน้ตกันหลบไปตกorganizers. It gives me an immense pleasure to be here today to give a keynote address this remarkable in science conference. Tapi mereka berapa itu lah? Kita kan nomor lucuan sih lah. Kan nomor lu terus sih kita lah kan. Mana?
Kan sana minit kata lah Oma.
Kan Jerman ini kan di Medan ini kan
Jerman. Oh, tihan kan roncan mo pangakalaw som. Ang je. Okay, mahian. Okay, mahi ani tok may vo insu. Dona na.
Karan ni kan ni arin timo. Signal ni minti boy la.
Oh, boy la tura ngay la. Oh. Alaw boy pa tut pon.
Alaw thau tro.
Oh, tidan kan nay to zel don niya.
Allah.
Kay mati kan impin no. Kan chan may don minyang oh. Hello and welcome to the second day of the Mizoram Science Congress 2020, which is conducted in the online mode. Today, we are very privileged to have in our midst one of the most prominent figure in the academic circle, as well as being a member of the current Legislative Assembly. Here with us, we have Professor Eflal Nunmoya, who really needs no introduction, as he is a very, very prominent figure here in Mizoram. So a little bit about the Professor Eflal Nunmoya. He finished his Master's of Science in Botany from the University of Nehu and PhD in the Department of Forestry from Mizoram University. He served a long career in the academic circle in Mizoram University, where he holds very important positions such as a professor in the Botany Department, as well as the Dean of Students Affairs. And now he is one of the key members in the current Legislative Assembly. So without further ado, I will request Professor Eflal Nunmoya to deliver the keynote address.
Okay. Thank you. Organizers of Mizoram Science Congress 2020. Especially Chief Scientific Officer, Mystic, and his the officials in the Mystic, and all participants of today's this Mizoram Science Congress 2020. It gives me an immense pleasure to be here today to deliver a keynote address at this remarkable session of Mizoram Science Congress 2020. And I'm also very honored to be a part of it. I would like to thank the organizers, Mystic, for organizing this program, which brings together academicians, research scholars, students, and all persons of scientific community to deliberate on sharing of information from various scientific researches in the state, as well as other parts of the country. So today's lectures and presentations will cover important disciplines such as life sciences, biomedical sciences, agriculture and allied sciences, and art sciences. So scientific researches, especially on our today's subject of interest, are comparatively new fields for the state of Mizoram. So we have recently held a science congress in the recent years. So where we have discussed recent development in different fields of science, especially for in Mizoram. So as we know, Mizoram has host a rich diversity of plants and is one of the Indo-Burma biodiversity hotspots. So the rich biodiversity of Mizoram is comprised of many important herbs and tree species that are used as traditional medicines. So similarly to many other regions of India, a good number of people depend on traditional medicines for treatments of diseases and ailments. So I have visited different parts of Mizoram in the recent years, the recent past, and I have observed that people heavily depends on these traditional medicines. So surprisingly, even many people residing in cities and big towns and in Mizoram, they follow the use of traditional healing system, and almost most of them would combine it with Western medications. So records of medicinal plants in Mizoram are mainly found in the form of books, articles, and through interviews of the traditional healers in different parts of the state. Recently, few research in environmental sciences and in botany have set light into the distribution of some medicinal plants in Mizoram, their botanical names and traditional uses. There are many more medicinal plants that are yet to be discovered and verified with the help of locals and traditional healers. So knowledge gained in the recent years pave ways for phytochemists and molecular biologists to study the constituents of individual plants and their bioactivities in different systems. However, systematic interventions in this area is still limited, and we are only at the initial stage in this subject. So one main reason that prevents the progress and expansion in the field of medicinal plant is the lack of systematic inventory system. Therefore, a structured and organized information system such as database, which is easily accessible, would immensely enhance our knowledge and augmentation of research in this area. I think you can hear me. Hello?
Yes, sir, we can hear you.
Yeah, okay, thank you. So this would benefit not only the scientific community, but the general public as well, and could contribute significantly to our society. And let's. I'm going to the botany and zoology stream, which some scholars will give a presentation today. I'm also very pleased with the work done so far in the field of life sciences, such as botany and zoology within the state of Mizoram. So new scholars have come up uh, with a new ideas, and they will. I mean, in future also, I hope they will take up a new research in this botany and zoology subjects. Identification of different plants, animals, and including mushrooms, fishes have been carried out, which provides both personal and societal gain. Many new species have been identified and described by our zoologists, especially from Pachunga University College and from MZU. So our professors and researchers have been working very hard. So in this zoology and botany, it is also very inspiring to know that research have been taken up in the field of mycology by some people in the Department of Environmental Science. So I also urge researchers to study species of plants, insects, animals, etc., at their genomic level. This will provide better and more in-depth information about the rich biodiversity of the state of Mizoram. So I'll just come to the agriculture and food production. So in terms of, Mizoram being a rugged mountainous areas and very steep slopes, it has the largest area under slope, more than 30%. So which is much steeper as compared to other parts of the state, the country. So as such, to increase the production of the state needs to explore. So to meet the ever-growing demand. So in this regard, there is a big challenge ahead of us for self-sufficiency in food crop production. So in this regard, crop production needs to be improved to increase the productivity and GDP of the state, and also increase the per capita income. So we have to focus and we have to help the farmers in remote villages to boost their income. So in terms of horticulture, high market value coupled by high nutritional value crops needs to be emphasized now. So for example, dragon fruits, which is a high value crop, a fruit. So it has now gaining popularity in the state and even other neighboring states. Increasing the productivity of such crops to an extent that they can be exported to other parts of the country, which would greatly improve the economy of the state and increase the GDP of the state, provided the government link secure market is available. So in both of the above cases, the marketing potential area for production in terms of geographical, geological, ecological, and climatic conditions through the use of modern technologies and GIS. So in this regard, the contribution of Mystic Science and Technology Department is remarkable. So they have developed a number of study. I mean, they have made a number of studies throughout Mizoram, different agroecological zones and geographical zones, climatic zones. So which, based on which, I think we can go for cultivation of some different crops based on a cluster mode. So in this regard, scientific intervention is very essential to improve or increase our food crop production. Lastly, I would like to say a few things on art science and its impact on the society as well as the environment. With it being a growing part of science in the last few decades, many inputs and research has been undertaken um, by different streams under the umbrella of art science, which proved to be very useful for the state of Mizoram. And I'd like to mention about some of our young geologists. We know that the recent earthquakes in the eastern parts of Mizoram, our young scientists, geologists have made significant contribution. And I'm very happy and impressed with the contributions made by our young geologists in explaining the details and fact of the recent earthquakes in Champhai district of Mizoram, which hampers the lifestyle, the life of many people in the affected areas in different ways. It clearly shows that their input provides not only the scientific knowledge on earthquake, but also comfort and but also bring comfort and peace to the people in the affected area. Apart from this, many other research and activities done by our young researchers in the field of art sciences and environmental sciences, like forestry, environmental science department, which are very useful. And I also wish that more research should be carried out throughout the state in the field of art science, which I'm sure will bring about prosperity and development to the state of Mizoram in years to come. So lastly, I am sure that the research findings, presentations, and discussions on today's topic will generate new ideas and hopefully result in at least some tangible information which may be compiled in a systematic way, which may be used for in future in different development initiatives of our government as well as private sectors. So with this, I end my keynote address and I wish everyone a fruitful and successful Mizoram Science Congress 2020. Thank you. Thank you all.
Thank you very much, Professor Efla Lunmoya, for giving us a wonderful insight and potential areas for exploration and research activities covering all the areas of agriculture, horticulture, biomedical and life sciences, as well as earth sciences, and how we can associate these research activities and the scientific interventions for the benefit of the society. Kanji nga Professor Efla Lunmoya ahun boy tak kara kanji nga aron tel thei sham sham hi kan lom ema hetyang ang mithiam science and technology lama shiat na nei zau le shiat na nei thu kanji nga aron tel thei hi science society science and technology society shang shang ami tile athok tu tile zir lai ten kan thamon pui ani kan lom sir.
Thank you. Hello.
For some reason, I forgot to introduce myself in the beginning of the session. So let me do it now. My name is Dr. Lalchandamitautong, and I'll be your host for today for all the technical sessions. So we have today in the morning session, agriculture and allied sciences for oral and poster presentation, as well as and in the afternoon, it will be followed by life sciences, biomedical sciences, and earth sciences. So for today's technical session, we have 4 expert members. Let me give them short introductions. We have Dr. S.T. Lazar Zovi from the Department of Environmental Sciences, Mizoram. Professor Devesh Walia from the Environmental Studies Department, Northeastern Hill University, Shillong. Dr. Laldusa Ngapatuao, Department of Pharmacy, Assam University. And last but not the least, Dr. H. Lalruatsanga, Department of Botany, Patsunga University College. I thank all the expert members for spending, agreeing to our invitation in spite of your very, very busy schedule. And I wish all the presenters for today's presentation a success. So we'll start with, we'll kick off now with the Agriculture and Allied Sciences oral and poster presentation. We have the first oral presentation for today, Si Zoming Sangi from Mizoram University. Si Zoming Sangi, if you are present, kindly share your slide and please go ahead with your presentation.
Yes, ma'am. Am I audible?
Yes, you are audible.
Okay. Greetings to our respected delegates and all the members present here today. I am Sizo Ming Sangi, Research Scholar, Department of Environmental Science, Mizoram University, and my topic is Assessment of Soil Organic Carbon and Available Nutrients from Secondary Forest to Oil Palm Plantation in Kolosip District, Mizoram. Introduction. Palm oil is obtained from the fruit of African palm oil tree, Elias ghanensis, and it is the most widely produced edible vegetable oil in the world. And the plantation of red oil palm began in 2004 and 2005 in Mizoram, and it is cultivated in Lunglei, Mamit, Serchip, Kolosip, and Aizol district, and the supervision and financial aid by Godrej Oil Palm Limited, Rushi Soya Industries Limited, and Foods, Fats, and Fertilizers Limited. And objectives: to evaluate the variations of some physical and chemical properties of soil from forest to oil palm plantation, and to recommend proper management practices. And materials and methods: selection of the sampling sites. 15 years old oil palm plantation owned by Mr. H. Lalramzawa, about 6 hectares located in Kamrang village, Kolosip district, and the secondary forest located about near the experimental plot, which is about 2.5 hectares. And soil collection was carried out in 28 February 2017 at 0 to 5 centimeter and 5 to 15 centimeter depth for 3 different slope percentage, namely the upper, middle, and lower. And sampling method: V method was used using the soil auger, and sterile plastic bag and insulated ice box was used for the sample transportation. And for our analysis of soil samples, it was carried out in the Central Lab ICAR Kolosip. And here are our methods we use for the analysis of our soil samples. Results and discussion. Here is a table showing the comparison of properties of soil from forest to oil palm, and our results are the mean of 3 replicates. pH. Soil pH declines significantly down the slope, as we can see here in both oil palm and forest. This may be due to the decline of exchangeable cations. And soil moisture content. Soil moisture content is higher in forest soil than oil palm soil, and the lower slope of forest has the highest soil moisture content. This may be due to the location of the slope, which is nearby the Tlong River. And the higher concentration of soil moisture content in the forest soil revealed that the presence of the thick layer or higher organic matter on the forest floor might have reduced the rapid evaporation of soil water. And soil organic carbon. Here we see a higher concentration of SOC in the forest soil than oil palm soil. This may be due to the higher clay content in the forest soil, and may also be due to the accumulation of organic matters over the years. And the less content of SOC in oil palm plantation may be due to the effect of land management activities under the plantation, And the regular clearing of the surface through weeding and removal of fallen leaves leads to low microbial activities. Available nitrogen. We see a higher concentration of nitrogen in the forest soil. This may be due to higher decomposition rate of litter fall, and the low concentration of nitrogen in oil palm soil may be caused by a number of fractions, such as the poor drainage and the water locked in the soil, and may also be due to the competition from aggressive weeds like the grass Imperita cylindrica. And available phosphorus. It is higher in the forest soil, which may be due to the rapid recycling of nutrients by decomposition and mineralization of litters. And the lower concentration of phosphorus in oil palm soil may be attributed to sandy soils that are more prone to moisture stress during the dry season, which will experience the additional problem in uptaking the vital plant nutrients. And potassium. The availability of potassium in soils is regulated by the content of organic matter decomposition as humus and soil pH. Thus, we see a higher concentration in the forest soil than the oil palm soil. And the lower concentration of potassium in oil palm soil may be due to low pH of the soil or sandy soils, which may cause leaching and drier conditions around the area. And the available potassium contents under the soils of the studied land uses were rated medium. Conclusion. Topographic position and ground slope accounts for variations in soil properties along the Ketina. There is the need to manage parts of oil palm plantation on different topographic positions differently, taking cognizance of variations in soil properties in order to ensure long-term sustainability. And our results suggest that conversion from forest to oil palm plantations can lead to the diminishing of SOC concentration and available nutrients, although this should be further validated with more studies, and this may have a negative impact on soil quality, the long-term productivity of the soil, and the global carbon balance. Last but not the least, with regards to soil fertility status, the soils in oil palm range from low to medium, and sustaining and improving the production capacity of agricultural plantations is therefore very important, and maintenance of the soil resources is a key issue for sustainable production. Thank you.
Thank you very much, Sizo Mingsangi, for your presentation. Now the time is open for questions from the expert members. Is there any questions that the experts would like to ask?
Okay, Ming Sangi,
Yes, sir.
even though I'm not in your line, I would like to know briefly from your studies, what is your overall suggestion about this plantation with this oil palm, how to perform this sustainable plantation of this oil palm, because it seems like it does have certain negative impact on the soil quality
Yes, sir.
or like that.
Yes, sir.
So how can we do oil palm plantation without, or maybe to rejuvenate soil quality with this oil palm plantation? Is there any suggestion you have?
Yes, sir. Look, we cannot like oil palm tell uh, to tell the truth, but for sustainable development of oil palm, it is suggest that we do not clear our virgin forest and use our fallow or fallow land for oil palm plantation instead of clearing our virgin forest, and then so that because our geographical area of Mizoram is not as large as other other cities or other countries, so it is purely suggested we don't clear our virgin forest and we use our fallow land, which are not used for other plantation crops, to grow our oil palm plantation. And this is a quite a controversial topic, as because the sustainable development of oil so it is quite hard, because even if we see some studies in the research article, we see many negative impact of oil palm plantation, because it disturbs the environment. So there is a much needed, more studies needed to be taken up, such as our studies which will be coming up in our journal papers, such as the carbon pools and stock, which will give a much clearer view of oil palm plantation. And from that studies, I think we can get a more clearer view of the oil palm plantation and suggest more
Okay.
better impact on the sustainable development of oil palm.
Yes, yes. Thank you. So, any idea? Do you have any idea what is the this yield of oil from this oil palm in the area you have studied?
Pardon, sir, I cannot hear you.
Uh, how how much is the oil this yield of this oil palm? Do you have any idea? You may not have. If you don't have, it's okay. I'm just curious whether the oil yield are quite good or not from this plantation sites you have studied.
Yes, sir.
It may be out of your topic.
Yes, we don't study the yield of the oil palm as of now, but from what our farmers said, that the yield is quite good, they say, but so it depends on, right?
Okay, fine. Yeah.
Yes, sir.
Maybe out of your topic. I'm just curious about it. Okay. Anyway, thank you. Good presentation.
Thank you, sir.
All right, thank you very much, Sizom Hingsangi. If there are no more questions, we'll move on to the next presenter, Irene Dalnipari. If you are present, please share your slide and you may continue with your presentation. Irene Dalnipari from Mizoram University. Is Irene present? All right, moving on, if Lalbiak Dika is there, we'll ask Lalbiak Dika to present his topic. Lalbiak Dika from Mizoram University.
Hello.
Hello. Do we have to share your slide?
Yes.
All right, we'll share your slide. Just hold on. All right, can you share, can you see your slide?
Not yet.
I can see your slide. Biak di ka ilang mutay law miya.
Một thầy chưa lâu.
Mo, nang man laptop ay hong thayem. Kay mani tuanin kan mutay to amo. Ka slide, ka screen na po alang ya. Oh.
Yes.
Okay. You may start when you're ready.
Ka share vele kher ngaim.
Ngay law, kan law share. Next slide law ti mayro. Oh, okay.
Okay. Yes. This is a poster presentation. Seasonal variation of soil nutrients in the paddy field of Changpai, Kolasip district. The objective is to investigate soil nutrient status like soil organic matter, nitrogen, phosphorus, potassium in the study area. Next slide. Yes. The soil sample was collected from 0 to 15 centimeter depth, and the total 10 samples were collected during before growing and post-harvest of the rice paddy. Here is methods. Here the observed data are analyzed by SPSS. See, and we can see from the table. And result. Soil organic matter was found increased toward the end of harvest. That is from pre-planting and growing to post-harvest. That might be due to increase in decomposition of plant materials. Other nutrients like total nitrogen, available phosphorus, and available potassium that was highest during the growing season. It is due to, it is because of all of the farmers applied chemical fertilizers in their field. Any question?
All right. Is there any question from the experts?
Yeah, I'll just ask one question regarding this soil organic matter. Your soil organic matter, it increases during the growing period. So before planting, it was 1.729. During the growing period, it was 1.98. Okay, so during the growing period, I don't think there was much decomposition. Decomposition will come after the harvest, I think, mostly. So how do you explain this increase in SOM?
There's a lot of weeds around the field, and they might not clear all the weeds after plowing.
Okay.
Yes.
So you think
before planting really means before plowing of the field.
Okay.
And data was collected during September and October.
Okay, so you think it's because of the decomposition of weeds?
Oh, yes.
Okay. And about this nitrogen, phosphorus, and potassium, you think it increases because they apply fertilizers, right?
Yes, yes.
Okay, so regarding that, do you think that your experiment, does it show how the paddy it affects the soil? Do you think it shows that because it is in an uncontrolled environment, you are applying fertilizers to that, so you don't know how much nitrogen is being applied. You can't know how much phosphorus or potassium is being applied. So does it give like a realistic outcome of how the paddy is affecting the soil?
I think Byagdika has lost his connection.
Okay.
We'll ask him to answer later on through WhatsApp. Okay. All right then. We'll move on to the next presenter. Poster presentation by Dal Nunmoya from Patsunga University College. Hello, Dal Nunmoya, are you present? Hello? I think maybe he has some technical issues. While we contact some of our members, we'll just have a short break. So kindly bear with us. All right, welcome back. We will now move on to our next session, the oral and poster presentation session for life sciences and biomedical sciences. We have our first presentation in this session from Purba Jyoti Deka from Mizoram University. Purba Jyoti Deka, you may now share your slide and start your presentation. Hello? Hello? All right, moving on. Is Benjamin Lalbiak Moya from Mizoram University present? Benjamin Lalbiak Moya.
Yes.
All right.
Can you
Yes,
hear me?
yes, I can hear you.
Okay.
So when you're ready, please start your presentation.
Mm, Can you see my screen?
Yes, I can see your slide.
Okay. My name is Benjamin Labiuk Moya from Mizoram University, Department of Environmental Science, and I am under the supervision of Dr. John Zautamzama. And our topic for today is morphological identification of mushroom from Polreng Wildlife Sanctuary of Mizoram. For mushroom are visible fungi with distinct copepods which represent the reproductive life cycle of Basidomycetes and some Ascomycetes. And mushroom gains more attention than the mycelium, which are generally found inside the soil or inside the decomposing matter. Uh, and mushrooms serve as a potential source for nutrition, medicinal, agriculture, and industrial application. Globally, around 40,000 mushroom species are known to exist, with only 1/10 taxonomically identified. In India, around 10% has been investigated. And in Mizoram, we have a number of studies done by different persons, but no studies on the Polreng Wildlife Sanctuary has been done till date. So the objective is therefore to attempt to explore and study the taxonomy and diversity of mushroom from Polreng Wildlife Sanctuary. Here in our objective, we try to identify mushroom of different species by observing morphological characteristics, and our study also aims to lay foundation for future studies as it serves a potential for nutrition, medicinal, agriculture, and industrial applications. The study site is the Poreng Wildlife Sanctuary, as we can see on the map. It is the northernmost wildlife sanctuary we have in Mizoram, which is located in the Kolisip district. For our methodology, we use the morphological analysis. As we can see on the right side of the slide, the different part of the mushroom. In morphological identification, we usually observe the visible macroscopic structure parts of the mushroom. In here, we have given the pilist, the hymenium, the annulus, the stipe, and the vulva. We also use microscopic method for the identification of mushroom by mounting a small portion of the mushroom on a stained slide mounted on a compound microscope with a small portion of the mycelium. Uh, by analyzing the morphological characteristic, we identify the mushroom by consulting appropriate literature. And in the next slide, I'll explain the different part of the mushroom. Here we can see the cap, the gills, the stipe, the hymenium, and the stalk. Uh, in mushroom morphological identification, the most important step is to record these mushroom when we collected them. As mushroom degrade very easily, it is very important to record this part at the beginning of the collection, preferably on the field. And on the next step, we observe uh, the spores and the spores under a microscope by mounting a small portion of the mycelium. On the right side, we can see the spores, which are uh, around 8 to 9 micrometer in size and are in spherical shape. And by consulting different literature, which I have given in the reference, we can tell that our mushroom sample is Amanita gemnata. And here are the species which we have identified using morphology so far. I will give a detailed pictures on the next slide. We have Amanita gemnata, Ammoderma rugosum, Arucularia cornea, Clavilopnesis lenticolor, uh, just Cobrinellas dissemantus, Dacrypinax, Spatularia, Dandelia quercina, Ganoderma apalentum, Lentinas sojorcaju, Lezites elegans, Microporus xanthopus, Phallus industiatus, Pinoporus coccineus, and Schizophyllum commune. And for our conclusion, the study find promising results on different type of mushroom inside Borneo Wildlife Sanctuary and can serve as a potential for food and other use. The study also lays a groundwork foundation for further studies, understanding the morphological identification of mushroom can be very useful even for the local people as it can help to distinguish edible and non-edible mushroom. Molecular analysis is also recommended to go alongside with the morphology as a number of species are very hard to identify morphologically and to also explore the genetic diversity of the mushroom. Nutritional analysis of mushroom is also recommended as it can serve a potential for food, medicine, and industrial use. That's all. Thank you.
Thank you very much, Benjamin Moyo, for your presentation. Are there any questions from the expert members? All right, if there are no questions, we'll move on to the next presentation. Let me invite Yang Samgon Gam Umbon from Ripans to do his poster presentation. We'll be sharing your poster from here.
I'll do it. Everyone uses Classroom.
Hello.
Hello. Yes, we can hear you.
Yeah. Okay.
Please start. Yes.
So my title is Determination of Nutritional Composition of Locally Produced Vegetables that are used in Mizoram, like in Northeast and other regions. So the vegetables I'm working on is Emoam dilbetum, Senegalia pennata, and Parkia timoriana. So here we determine the nutritive values like the protein, carbohydrates, fats content of these locally used vegetables, and we concluded that the nutritional value which are beneficial for consumption. So here at first, the methodology of this experiment is we have determined the protein content by using the JEDEL method of protein estimation, where we used where there are 3 units: the digestion unit, the distillation unit, and then we have undergo titration for determination of protein, and then carbohydrate. We went for the Entron method, and we also determined the ash and moisture content of the vegetables. So moving on to results. So after the determination of the calorific value, it is found that Senegalia pennata has more calorific value than the other 2 vegetables, and although all the 3 vegetables have good amount of calorific value in them, Senegalia pennata achieves more calorific value, which has more health benefits for the consumers, and we can conclude that the vegetables that we use for experimentation and determination of calorific value shows good result of nutritional properties, and its consumption may lead to various good benefits for health. Thank you.
Thank you very much. Are there questions from the expert members? Any questions for Yang Sangam?
Okay. Yes, Sangam.
Yes,
So
sir.
you have performed some most probably a little bit of preliminary studies in a sense. So anyway, very good work you have done. It's so these nutritive values what you have determined so far, are they biologically accessible? Are they biologically accessible kind like what we can digest and what we can use for this nutrition in human? Biologically accessible, are they?
Yes, sir. They're a very good source of nutrition, and like they have many traditional medicinal use. Like that I do is also used for treating hypertension and even diarrhea. So I guess we can use it for our health benefits as well.
Okay, good. Because why I'm asking is that because in the lab we may be able to find out several qualities like calorific value, etc., etc. But when we actually consume it in human condition, are they actually means translated into human consumption situation? In that sense, I was asking.
Yes, sir. They do. Like here, we determine the total nutritional value of the particular sample we use, so we can get almost accurate percentage of nutritional value content of that sample.
Okay. Yes, yes. Good. Okay. Anyway, very good. Very good work.
Thank
Yes,
you.
Sangam. Yes, Sangam.
Yes, sir.
Could you hear
Yes, sir.
me?
Yes, sir.
It's a good study and very applied
Thank you, sir.
to our it's very applied. So what is the way ahead? Do we simply recommend those particular vegetables to all the household?
Yeah.
What is the way?
Excuse me, sir. Your voice is cracking. I'm not getting you.
Where do we go from this? And further to the
No, like the main aim of this study was to make the people aware, like are they really getting the nutritional value, like the nutrition from the foods they're consuming. So like for that we have determined the nutritional value of those locally produced vegetables, so that like the common people will be aware of it, and they can consume it up to their will.
yeah, without hesitation. They'll feel they'll be confident in eating in having those vegetables. And I suggest we go for yet another common vegetables available in a market, so that the people can have a faith and confidence with
Yes, sir, we can do that.
the local available vegetables. It's commendable. Thank you.
Yeah. Thank you, sir.
Yeah. Okay.
Thank you very much. If there are no more questions, we will move on to the next presentation, poster presentation by Sidal Sangswali. Sidal Sangswali, when you're ready, please start your poster presentation.
No, they will share it.
Just talk.
Do we need to share your slide?
Yes. Yes. Yes, Miss.
Okay. Okay. All right. Can you see your poster?
Yes. Yes.
All right. You may start when you're ready.
My name is Sira Sangwali I'm from Ripans, and I'm going to present a poster presentation of determination and comparison of nutritional composition of various rice samples found locally in Aizawl, Mizoram. And the main objectives of this project is to determine the calorific value of these 8 rice samples, which are basmati rice, arham, common rice, boiled rice, Ido, Fazai, BBT rice, and SNT rice. And this basmati rice is mainly from Punjab and Haryana, and arham is mainly transported from Andhra Pradesh, and and common rice from West Bengal, and this boiled rice is mainly obtained from this is from Assam, and this Ido and Fazai is from Mizoram. We call Bukan, and boiled rice. This BBT rice is from I think this is from Andhra Pradesh, and SNT rice is from Gujarat. And in the introduction, we have the scientific name of rice, or Rhiza sativa, and this rice is mainly consumed by most of the people, and it provides bulk of calories for animals and humans. And also that rice contain many nutrients like carbohydrates, proteins, fats, vitamins, and minerals. Can you go to the next slide? And this is the methodology: carbohydrates content, fats content, protein content. From these 3 procedures, we can calculate the total calorific value of all these rice samples. First, we calculate the amount of carbohydrate in the sample. Then after that, fats content, then followed by protein content. Then after we get the result, we have this total calorific value from the following formula. We sum up the calorific value of carbohydrates, fats, and proteins. And these are the results obtained for each rice samples. And the last one is the discussion. And from the discussion, we conclude the bar graph, and we can conclude that boiled rice has the highest calorific value, followed by common rice. And also these are they are cheap in price and also convenient to consume and produce sufficient amount of calories. This is all about my presentation. Thank you.
Thank you, Sira Sangwali. We'll now take questions from the expert members.
Okay, Sangwali.
Yes, sir.
You have told that boiled rice is highest calorific value.
Yes, sir.
Okay. Is it boiled rice comes from some other place or from Mizoram you have obtained?
No, this boiled rice is from mainly Assam, because we got it from canteen, Zemabo canteen.
Okay, near Ripans. Okay. So I was just wondering whether the means the age or how long it has been stored to these samples, whether it will have any influence on their calorific values. But anyway, is there any explanation you have why it contains a higher calorific value than other samples?
Because this boiled rice, as the name implies, it is there is a slight modification here. Like first they soak the rice, then they boiled it, then after that they dried again, and also it absorbed most of the nutrients. That's why it has high calorific value.
Okay. Okay. Thank you.
Yeah, I have not a question actually, but I'm a bit surprised that boiled rice has the highest total calorific value. And I'm looking at the carbohydrate content, and I thought boiled rice will have the lowest carbohydrate content because it's recommended for those people with diabetes. So I'm a bit surprised that it has a very high carb content.
Because it contains other nutrients, like the total fat and protein percentage is high. No, that's why from the because we calculate the total calorific value, so it all matters from that.
I think maybe what matters is also the glycemic index, right?
Yeah.
All right. Thank you very much. We'll now call our next presenter, Dr. T.C. Lalshat. We will be sharing the slide from here. All right. Can you see your slide?
Yeah, yes, ma'am. Yes.
All right. Please start when you're ready.
So good morning, respected teachers, respected experts, and all the audience. Today, my title is Determination of Protein Contained in Different Marketed Honey Sample, Dabar, Epis, Rasna, and Local Honey by using Zeldan method and their comparative studies. And my co-author are Dr. Rosangi Sumbeni Mayato Sachu. So the introduction is, as we know, all knows honey is natural product from nectar of flowers by honeybees, Apis mellifera family is Apidae, and then honey is the only insect-derived natural product, and it was nutritional, cosmetic and therapeutic, and it has also some industrial value. The water gravity of honey is between 0.56 and 0.62, and its pH value is 3.9. Honey is utilized as natural sweetener from ancient period since it has high level of fructose, as honey is 25% sweeter than tablet sugar. Then moreover, the use of honey is in beverage is also becoming popular. So biological activity of honey. So these are the reported activities: antioxidant activity, hypotensive and blood regulating action, anti-inflammatory and immunomodulatory activities, reducing it will reduce the duration of diarrhea. It will prevent the acid reflux, and also it will relieve the cold and cough symptoms, and effect of honey on immune system is also very prominent. So objective of this presentation is to determine the protein content in the different marketed honey sample. Like we have taken 4 sample, so we determine the protein by using Zeldan method, and then we compare their content. Next slide, ma'am. So the method what we use is Zelda method. So the method is consisting of three unit. So here we can say that the first unit is digestion. Sample whatever we have selected were digested with concentrated sulfuric acid in the presence of catalase, and then distillation unit is there, and it was done in the presence of 40% sodium hydroxide and 4% boric acid and distilled water, and then titration. Distillate obtained was titrated against 0.1 molar hydrochloric acid. So this Zelda method is done using the instrument Zeltron. So Zeltron is available in our institute, so we perform this experiment, and then here result we compare by, so the proteins content was calculated by using the formula. This is the working formula given. The percentage of crude proteins available in the sample is calculated by 14 into normality of acid, then it is multiplied by actual titration value into the power factor, and then it is divided by the weight of the sample into 1000, and the whole is divided by 100. So it is found out that the percentage of crude proteins from Dabur, Apis, Rasna, and local honey was found to be 0.65%, 2.87, and 0.79, and 1.63 respectively. So here, what we have found is that Apis is having the highest protein content, and the second one is local honey, and the third one is Rasna, and the fourth one is Dabur. So discussion, protein is essential nutrient in the body that is utilized for many functions. As we already said, the percent of protein intake can be determined through their rice standard, and then with the result obtained from the comparative analysis of protein content in the selected honey sample, Apis honey sample is recommended as compared to other honey. Also, it is recommended that future research should be carried out on the approximate nutritional analysis and mineral content of the honey sample. Thank you.
Thank you very much, TC Lashatpuri. We'll now take questions from the experts.
Okay, Madam.
Yes,
thank
sir,
you. Thank you for your presentation. What I would like to know is that you have determined protein content based on nitrogen
yes.
determination. So have you considered any non-protein nitrogen content in the samples so that you got the real proteinous nitrogen?
Sir,
Is there any
actually,
chance non-protein nitrogen to be present in the sample? Is there any chance?
yes, sir, it is there, but actually, due to the shortage of time, like we could perform only very small experiment this time. So
Okay.
actually, the estimation of the some other minerals also can be evaluated, but sir, due to the shortage of time, we could not perform. Only this much can be performed for this particular presentation, sir.
Okay, so whatever the nitrogen you have found, you have considered it to be part of the protein.
Yeah, yes. So we regret for the insufficiency of time. Yeah. Okay.
Yeah. Okay. Thank you. Good work.
Okay. Yes. Thank you, sir.
Any more questions? All right. If there are no more questions, we'll move on to the next presenter, Jiko Gogoi from Optometry Association of Assam. Jiko, are you able to share your slide from your end?
No, ma'am.
Okay, we'll share the slide for you, okay?
Yeah, thank you so much.
All right. Are you able to see your slide?
No, ma'am, not right now.
Okay. We have shared the slide. I can see it on my screen. Are you able to see it on your screen?
No, ma'am. Sorry, I can't.
What you can do is, since this is a poster presentation, you can present from your end, and we'll just move the slide as you present along. All right? Carry on.
Okay. Thank you so much, ma'am. So, very good afternoon, ladies and gentlemen. My poster presentation is on cosmetic rehabilitation of one-eyed patients using lightweight prosthesis. So, everyone, we know that the eyes, they don't only play the role of a sensory organ like we can see with the eyes, but apart from that, eyes also play a very important role in the physical appearance of a person. So there are various medical conditions like cancer and other ocular infections that happens in the eye, for which reason we need to remove the eyeball, okay, to save the patient's life. Like if a kind of tumor or malignant cancer develops in the eye, then it can move to the brain through the optic nerve And so in order to save the life of the patient, we need to remove the entire eyeball. So after the removal of the eyeball, the facial appearance looks very disfigured, and the patient lacks in self-confidence and self-esteem. And this is more crucial in terms of kids, because in small children this type of cancer is very much common. It is called retinoblastoma. So after removal of the eyeball, the child gets socially aligned from others, alienated, and they don't feel like going to school and meeting new people, and as a whole their personality and their educational development stops. So what I have done, I have taken the impression of the empty socket after removing of the eyeball with the help of some materials which are used in dentistry, like what we used to make the um, teeth and other implants. So with that material, I have taken the impression of the socket, and I have tried to paint the exact features like the other eye, like the black portion, which is also known as the iris, and the conjunctiva, the white part. Even I have included the blood vessels also with a special kind of material and paints. After which, if we give a clear coating, like a plastic clear coating on top of that PMMA implant, that it looks like the other eye. And after proper insertion of the eye inside the artificial eye inside the socket, the patient looks very much presentable, and it almost mimics the other eye in terms of movements and in terms of appearance. Even the structure of the eyelids also stays same, because the space is not empty inside and it is filled with the implant, which I have painted and matched with the other eye. So before, what they used to do, they used to make this artificial eyes, the prosthesis, with the help of very low grade industrial plastics and other stone materials. So they used to say like stone eyes in early times. So that tends to become very heavy, and it also doesn't match with the other eye. Apart from that, due to the weight of the prosthesis, the lower lids and all those becomes disfigured in due course of time. So what I have done, I have made the same thing with the help of polymethyl methacrylate. This is known as PMMA. So the white PMMA I have used as the implant, and I have painted on that, and I have given a clear PMMA coating, the clear plastic coating on top of that with the help of a polymerizer. And after insertion, the eye looks like the other eye. So what it does indirectly, the person looks presentable in front of other people, and as a child there is no hesitation in the child in moving out to school and mixing and playing with other kids, because he thinks, or he or she thinks, that she is not cosmetically and physically different, looking different than the other kids. So as a whole, it also helps in the development of the mental and the personality development of the child. And as the prosthesis is very lightweight, so it is very much comfortable for the child to wear, and he can take care of it by himself after proper instruction and guidance. The eye can be removed only once in a month, at least for 3-4 minutes, and you don't require any very high kind of maintenance. That can be cleaned with any kind of disinfectant soap and can be easily inserted and removed. So I think this is going to help a lot of one-eyed and patients who have lost their eyes of any age, and I think this is going to help in future. Thank you so much.
Thank you very much. Are there any questions for Jiko Gogoi from the expert members?
I have one question, Jiko. It's
Yes,
the
yes, sir.
concept is good. The concept is good.
Yes, sir.
What you have done is to help the patient. But what about the
Yes, sir.
biocompatibility of your materials that you
Yeah,
use? Have you
yeah,
tried or have you
yes, sir.
tested any
Yes.
compatibility with? Okay.
Yes, sir. I have already fitted around 150 patients with this material, and as PMMA is a biologically inert material, so it doesn't react with any tissues of the body, and it has got no infections. I have studied this in last 6 months, and I haven't found any kind of contradictory result in this.
Okay, before you use on human, have you tried any on in vitro models or any animal models, whether they have any reactions, interactions?
Yeah, yes, sir. There actually the whole idea came to me by seeing one of my pet dogs. The dog he lost the eye due to an injury, and after proper healing of that socket, what I did, I tried taking impression of the dog with this material, and I have closely studied it for last 6 months, and I have seen that he was doing well with that model. That's why I tried it in a couple of patients, and it was turned out quite well.
Okay. Okay, fine. That's good. Okay. Because I'm thinking about the safety and the approval that may be necessary to be used on human. In that aspect, I
Yeah,
was
no, actually, sir,
wondering
yeah,
about.
actually, yes, sir, the material that I have used, the PMMA, the PMMA is already FDI cleared, and it can be used as implants in other parts of the body. And there are
Yeah,
some
it may
implants
be,
which they,
yes. Uh-huh. But it also, the way you prepared and the way you synthesized and all those may also influence, there may have some influence. So those parameters
yeah,
may also may be necessary to
yes,
study.
sir,
That was what I was thinking.
yes, sir,
Okay. Anyway,
yeah,
yeah,
you
the
are right, sir.
intention is very
Hmm.
good. The concept is good. The concept is very good.
Yes, sir. Actually, the materials are already used in dentistry, which is used in the mouth. So I thought that the same material can be used in the eye socket also, because that is also like a cavity. So thank you, sir.
Okay.
Jiko,
Okay. Fine.
hello,
Yes, sir.
Jiko. Oh,
Yes, sir.
are you a medical professional, by the way? What's your
Yeah,
background?
yes, sir. Yes, sir. Sir, I have done masters in optometry, which is
Okay,
related with
okay.
primary eye care.
Oh,
And
oh,
I have also done a special course in anaplastology, which is like making artificial nose, ears, and other parts of the body with silicone.
okay. That's great and fantastic. Good to hear that. So our
Thank you, sir.
priority is safety of our human being, our fellow beings.
Right, sir. Right.
So
Right, sir.
being basically an optometry master, yeah,
Yes, sir.
that your patients are in a safe hand.
Yes, sir.
It's good
Yes,
to
sir.
see, and like, mm, how long can the your patient make use of this?
Yes,
How
sir.
long do this material last?
Sir, the polymethyl methacrylate PMMA is non-degradable material. Okay, sir. So till now, I have
Mm,
already seen patients wearing for 2 years at a stretch without
yeah,
any
because
kind of
why I
complication. Yes.
yeah why I asked
Yes, sir.
is because your patient,
Yeah.
be it children or
Yes,
the
sir.
youngsters, if they
Yeah.
keep on growing
Yeah.
proportionately,
Yes. Yes, sir.
that
That's
that needs to be updated
yes.
or reshaped.
Yeah. Yeah, yeah,
Yeah,
no, sir, that has to be updated. That's a very good question. Yes, sir.
right,
Because
right,
as the child will keep growing, the
right.
sockets will also keep growing. So at least after a couple of years, at least after two years, we need to take a fresh impression, and we need to see is there any difference in the size and
Right.
shape. So if there is any change in shape and size, we need to modify it
Yeah.
accordingly.
All right. Thank you very
Follow
much.
up after a regular interval
Yeah.
of time,
We
and
need to
then
call the patients for follow-up after every six
okay,
months. The patient will keep coming to me after every 6 months. So I need to see whether the implant and the eye is getting any loose fitted anywhere or else. And I need to keep assessing him in a regular manner, at least after every 6 months.
All right. Thank you very much, Chico Gogoy, for your presentation.
okay, yeah.
Are there any
Yeah,
more
thank you, ma'am.
comments or questions? If not, we will move on to the next presentation. We will now request Purba Jyoti Deka to present his Purba Jyoti Deka, can you hear me? All right, we'll share the slides for Purba Jyoti Deka and then he can start when he's ready, right?
Okay, yeah. Hello, am I audible to everyone?
Yes, we can hear you.
Okay. Okay, hello?
Yes, hello.
Yeah, okay, yeah.
Please continue. Carry on.
Okay, good afternoon, everyone. My name is Purvojyoti Deka. I'm a PhD research scholar, Department of Biotechnology, Mizoram University. And today my topic of presentation is on diversity and succession of microbiota during fermentation of traditional foods of Mizoram, Northeast India. Please, next slide. Yeah, we all know about probiotic and the importance of probiotic in day-to-day life. So probiotics, they are the live microorganism which, when we administer in a small adequate amount, they confer some beneficiary health effect into the body. So we know that probiotics, they have very health benefits, like they boost our immune system, they help us in mental health, some good bacteria, weight loss, heart problem. They have many advantages as a health beneficiary, like anti-diabetic property, they have antimicrobial, anti-cancer. And to be a probiotic, they should have some properties, like they should be very non-pathogenic, they should be non-toxic, they should survive the passage through the digestive system, they should attach to the intestinal epithelia and colonize, they should maintain a good viability with the host, they should be anti-inflammatory, anti-mutagenic, and immunostimulatory, and they should be capable of exerting some beneficiary effect on the host. Next slide, please. And most of the probiotics food we have found from the fermented foods, and we know fermentation is an ancient method of preservation of foodstuff all over the world. And we know in Northeast India we have different communities of people, we have different ethnic groups of population, and they used to prepare and consume a variety of necessarily fermented foods and beverages. But till now, there are very less known about the microbial community and succession of such traditional foods. Therefore, we have done a survey inside the Mizoram, and we have found that most of the Mizoram people, like we can say the maximum number of people, they used to have some fermented foods like fermented bamboo shoot, which is called as dried sugar, fermented soya bean, fermented soybean, and these are being consumed in a higher amount. And you collected a sample according to the day of fermentation. Like we have observed the people, they used to have a third day of fermentation, fifth day of fermentation, and 7th day of fermentation. Therefore, we collected a sample according to the day of fermentation, and we have done their microbial community composition analysis along with the proximate nutritional analysis. Like we have observed the carbohydrate content, protein content, vitamin, like moisture content, ash content, etc. Next slide, please. So after collecting the sample, we have finalized our objective to investigate the microbial communities of the traditional fermented foods of Northeast India, especially from Mizoram, to compare the microbial communities which are prevailing in these different fermented foods, and to find out some probiotic potential through metagenomic mining and exploitation of those probiotic strain as a potential candidate for commercialization. So after collecting the sample, we have done the DNA isolation, and we have done the 16S rRGNA Illumina sequencing using bioinformatics tool. Along with that, we have done the nutritional analysis for different parameters, like I have already mentioned: carbohydrate content, protein content, fat content, moisture content, ash content. We have done about all these parameters. Next slide, please. Okay, so after doing all the microbial composition analysis by using this Illumina sequencing, we have found that lactobacillus, they're highly dominant in all the fermentation day for the bamboo shoot. Like we have found lactobacillus, they are dominant in 3rd day, 5th day, and 7th day of fermentation in the bamboo shoot. And in case of pork fat or the saum, we have found Clostridium sutrella lactobacillus, we have found dominant in all the fermented day. And in the case of Beckham, we have found Staphylococcus and Bacillus genus. They are more dominant in all the fermentation period, like 3rd day, 5th day, and 7th day. Next slide. Okay, well, after doing all this analysis, like we have found in the phylum level, the firmicutes, they are 72 to 72.87% dominant, proteobacteria 25.6%, and actinobacteria, they are 1.29% dominant in all these fermented food sample, along all the fermented their fermentation. Next slide. Next, please. Yeah, so at the genus level, we have found Lactobacillus 91%, Clostridium 72%, and Staphylococcus 52.36%. They are dominant in all the fermented food sample like bamboo shoot, pork fat, and soybean respectively. And interestingly, like all the genus diversity of all the three types of fermented foods, they are found to be found to be very diverse with the dominance of different species. They're differently dominant in each of the fermented food. Next slide. So we have done the comparison study for all the three fermented foods. Like we have found in fermented bamboo shoot, we have found in third day of fermentation. Third day, we have found like lactobacillus, they are more dominant in all the day of fermentation. Third day, fifth day, and seventh day. And in case of fermented pork fats, we have found like Clostridium, they are dominant in third day, fifth day, and seventh day. And in case of Bekam, the fermented soybean, we have found that Staphylococcus, they are dominant in all their fermentation. Like in each day of fermentation, like third day, fifth day, and seventh day, we have found there are different dominance of like genus or phylum. Next slide, please. So this is coming to the approximate analysis. I already mentioned about that what parameter we have done, like carbohydrate, protein, fats, group fiber, moisture content, ash content, and calorific value. So as per the day of fermentation, the parameter of nutritional this nutritional parameter, they are different. Like in case of fermented bamboo shoot in the third day, and in case of fermented bamboo shoot fifth day, the variation happens due to the this nutritional parameter factors. So we are still having some correlation study among these all the nutritional factor and microbial communities for each of the fermented foods still going on. Next slide. So this is since my work is still going on, so these are my conclusion and some future studies we are still having. Like this study is, if I'm not wrong, like it's the first report from Mizoram state of India, like about this all the fermented foods. And interestingly, all the three samples show the difference in the dominant species, indicating that the microbial communities depends on the process of fermentation as well as nutritional parameter of the food. Because we have observed like the community of people, they should prepare the food in different ways. So due to the mode of preparation, the parameter also changes, either in nutritional parameter or the microbial composition. And this study indicated that the most of the food sample, they contain some microbial species that may have probiotic potential. Since we already found lactobacillus as a dominant in all the fermented food samples, so there is more possibility to have some probiotic potential like lactic acid bacteria. So from all the three fermented food sample, I have already isolated more than 200 bacterial stain. And for the probiotic screening, I am still doing for the probiotic staining. So if from all the three fermented food sample, if I can have some good probiotic strain, that will be more useful for the future. Like we can use it for commercialization. So this work will be useful to the consumer by providing some essential and important information on the benefits as well as potential health risks associated with the traditional foodstuff. Next slide. So I would be like to thankful to my supervisor and to my co-supervisor and DBT for financial help. Thank you so much. This is all about my presentation and my research work that I'm still going on. Thank you.
Thank you very much, Purvajyoti, for your presentation.
Yeah. Okay.
We will now open the time for comments or questions from the expert members.
Okay, sure.
Purba?
Yeah, yes, sir.
I have one question, even though it's not your objective. Your objective is
sir.
towards, I think, probiotic aspect.
Yeah, yes, sir. Yeah.
But since you have identified many microorganisms,
Okay,
so
sir. Yeah.
I'd like to know some insight from your studies. What about the pathogenicity possibility of these pathogens present in the samples? Is there any
Sir,
pathogenic?
sir, there is possibility, especially in case of Bekang, this fermented soybean, because we have observed in Mizoram, most of the people they used to get poison. Okay, most of the people they used to get poisonous after having this Bekang. It is due to the day of fermentation. Like people they used to eat after like more like 7 day also sometime after when they used to have after 7 day of fermentation, like they used to have some poison because after that some pathogen used to grow. So while doing our this metagenomic analysis, we have found like Staphylococcus. We have found Staphylococcus dominant in the fermented food, especially like 7 days. So this there is some more possibility. We are still doing our work. We'll still further we'll go for this pathogenesis test, but there is possibility to have some pathogen if we have the food some delay period of duration. Like in the early period of fermentation, if we have the food, then there is no possibility of having pathogen. But later period of fermentation, there is possibility. So we are trying to
Okay.
include some pathogenetic work also further.
Okay, very good. So the longer it takes,
Yeah.
that means the more chances are there that
Yes, sir.
some
Exactly, sir.
pathogens will
Yes,
be
sir.
entered.
Yeah.
Okay.
Especially in
Okay.
case of fermented background, we have observed like some group of Staphylococcus. We cannot say whether it's directly pathogen or not. We have to check the pathogenicity, but there is possibility of having pathogen in further day of duration of fermentation.
Okay.
That's
Okay.
why we took the sample according to the people they used to have second day, third day, and fifth day like that.
Okay. Okay, fine. Thank
Yeah.
you. Thank you. Good work.
Yes, sir. Thank you.
Are there any further questions or comments from the expert members? If not, we will thank Purbajoti Deka,
Yeah, thank you so
and
much. Yeah.
we'll call the next presentation, who we missed in earlier due to technical issue, is Irene Lalipari present. Irene, can you hear me?
Hello.
Hello.
Am
yes. Yes, we can hear you. So our next presenter is Irene Lalipari from Mizoram University. We'll be sharing your slide from our end. Okay, Irene. All right, sharing your slide. Are you able to see your slide on your screen? Hello, Irene. Irene, can you hear me? Hello. Yes.
Am I audible?
Yes, you're audible. Are you able to see your slide?
Yes, Miss. I can see my slide. Yes, thank you.
All right. So you may start. We'll control your slide from here. Okay.
Okay, ma'am. Thank you. Thank you so
All
much.
right. All right.
Good afternoon, everyone. My name is Irene Lalipari Selete. I am a research scholar in the Department of Environmental Science in Mizoram University. The topic which I wanted to present today is assessment of climatic variables on the yield of maize in Mizoram, India. Next slide, please. Agriculture can be affected by climate change in numerous ways. Firstly, the quality and quantity of crop production, and then agricultural practices through the change of water use and agricultural inputs such as herbicides, insecticides, and also due to fertilizers. In addition to this, there are also the environmental effects in relation to the drainage and erosion of soil and the loss and gain of cultivated land and land use. the world population is currently 6.1 billion, and this is expected to rise to 9 billion by the year 2050. If we look at the population of India, we will soon become the most populous country in the world due to the advancement in medical facilities, in an increased life expectancy. So we are going to be faced with a great challenge on the agricultural sector. So in order to combat this increasing population growth and then the strain which it puts on the agricultural sector, there is a need to find a solution, or should I say a particular a crop which we can use to reduce the possible food shortage. So here I want to introduce the cultivation of maize, because of its ability to withstand a wide variety of climate, soil, and management practices. So the choice of crop which I would like to mention for sustainable development would be the maize. So for the introduction, I want to the urgency to cultivate crops that can withstand the changing pattern of precipitation, flood, and salt-resistant variety. Next slide, please. So my objective for my study is to determine the relationship between rainfall, humidity, and yield of maize using regression and correlation. Next slide, please. The study area which I have selected is Mizoram. Out of the total geographical area in Mizoram, vegetation accounts for 75.56%, which is approximately 15,935 square kilometers. There are three agroclimatic zones which I wanted to mention here: humid mild tropical hill zone, humid subtropical hill zone, and humid temperate subalpine zone. The rainfall which we receive is mainly from the monsoon season, and the mean summer and winter temperature which we experience within the study area ranges from 24 degrees Celsius to 30 degrees Celsius during the summer, and 10 degrees Celsius to 20 degrees Celsius during the winter temperature. Next slide, please. So for this study, the data which I've collected were compiled and analyzed to determine the impact of rainfall and humidity and the production of maize within the study area. Next slide, please. This graph represents the relation between humidity and the yield of maize for the last 33 years. Yield of maize was taken as the dependent variable. So we can see that there is a 1% increase in the yield of maize with an increase of 5% humidity. Next slide, please. When rainfall was taken as an independent variable and the yield of maize was taken as the dependent variable,
Hello, Irene, we are unable to hear you. Hello?
Maize yield by 14%. Next slide, please. Represents humidity and yield of maize, and moderate correlation between rainfall end yield, while end yield of maize. Next slide, please. In Mizoram, there is a lack of scientific research and observation, which can be attributed by the remoteness, extremely difficult terrain, as well as direct field observation. Availability of organic matter as well as the variety of maize cultivated could have impacted on
Irene, we are unable to hear you. Hello, Irene, are you still online? Hello? Okay, I think Irene has faced some connection technical issues. So I think anyway, she was able to present till her concluding slides. So we'll now break for about 5 minutes till we move, and then when we come back, we will start the session for Earth Sciences. I request you all to kindly join as we come back in a short while. Thank you very much.
Well,
Hello and welcome back to our next session. We will now start the Earth Sciences session. We will first call Mizoram Thara from Mizoram University to present his topic. Is Mizoram Thara available? Can you hear me? Hello?
Can you hear me, ma'am?
Hello? Yes.
I'm here.
All right. Do we need to share your slide or are you able to share it from your
No,
end?
I will share from my end.
All right, all right. All right, carry on. Start when you're ready.
Okay, thank you. Can you see my slide? Is it visible?
Yes, yes, it's visible.
Okay, thank you. I want to thank the
We lost your voice. Zoram, yes, now we can hear you. Yes.
yeah. Excuse me, ma'am. 1 minute.
All right, no problem.
Then
If you want us to share the slide from here, we can do it.
Okay, ma'am. If you can share, I have some technical problem.
All right. Hold on. Are you able to see the slide?
Yes, ma'am.
All right. Carry on.
My name is Dr. Mizoram Thara, Assistant Professor of Government Residential Science College. First of all, I want to thank the chairman and the organizing committee. Yeah, today I'll be presenting on provenance studies of. Ma'am, the slide is going. It's okay. Today I'll be presenting on provenance determination of Middle Buban sandstone of Suma Group, Mizoram. Uh, the study area. Next slide, please. The study area lies in the eastern part of Aizawl, that is the eastern flanks of Aizawl and Decline, and it falls in the survey of India topo sheet number 84A10, and it's the road section from Moalpui to Falkland, that is the World Bank Road, and samples are collected from this area only. Okay, next slide. The aim of the study is to reveal the provenance of Middle Buban sandstones of Suma group exposed around Aizawl city. Since Aizawl city, it falls in the Suma group of Buban formation, and mainly the rock comprises of sandstone, shale, and siltstone alternation. The methodology include the collection of samples from the study area, and after collections, the rock sample is soaked with water for 2 days, and after soaking, it is being grinded into pieces, and after the grinding is finished, heavy minerals are being separated by funnel separation method using bromoform, which have a specific gravity of 2.89 gram per centimeter cube. And in this funnel separation method, those light minerals are being floated while the heavy minerals sank at the bottom, and we separate the heavies by tapping off the heavies from the funnel. And after separation, it is being washed with acetone, since bromoform is carcinogenous liquid. So it is being washed with ethyl alcohol so as to remove all the bromoform left over in the samples. And after the separation is finished, it is being observed under petrological microscope. That microscope is a Leica DM2700P with a high resolution camera and Leica image software. Next slide. This is the field photographs of the study area. A is the sedimentary succession exposed along the road section of World Bank Road, that is below this A is below the petrol pump near located at Falkland, and B is the quarry site near the Falkland Park. Okay, next slide. The heavy minerals was carried out using selected 10 samples out of the 31 samples collected, and various heavy minerals were observed, and after observation, the optical characteristics are being studied, and these heavy minerals is used for determination of provenance, explained by Carven, 1971. So in the study area, the heavy minerals identified under the microscope comprises of sillimanite, olivine, zircon, staurolite, hornblende, garnet, chloritoid, enstatite, tourmaline, muscovite, kyanite, bitite, monzonite, rutile, chloride, diopside, glaucophane, magnetite, and hematite. Among these heavy minerals, magnetite and hematite, these opaque minerals are very common and their population is very high. Okay, next slide. These are the heavy minerals identified under petrological microscope. So we selected the heavy minerals for the provenance studies because these heavy minerals are very resistant to weathering, and they can depict the origin, the source rock origin. They can depict the source rock of the sedimentary rock. So the identified heavy minerals include sillimanite and olivine. These two shows the high-grade metamorphic to basic to ultra-basic igneous rocks origin, while zircon, that is E to J in the photographs, shows acidic igneous rocks. Enstatite shows U to X shows basic to ultra-basic and high-grade metamorphic rock. Okay, next slide. This is the heavy minerals identified so far. As we can see, tourmaline A to C, it shows a perfect euhedral shape. That means it is an igneous origin. While kyanite and kyanite shows medium to grade metamorphic rocks. The glaucophane shows the blue stitch metamorphic origin, while hematite shows igneous and sedimentary origin. Okay, next slide, please. On the basis of the nature and mode of occurrences of the identified minerals, it is concluded that the provenance of the investigated Mizoram sandstones are cosmopolitan in nature. That is, it is a mixed environment where low-rank to high-rank metamorphic rocks, acid to mafic igneous rocks, pegmatite, and reward sedimentary rocks are the provenance of the studied Middle Buban sandstone. Thank you.
Thank you very much. Are there any questions from the expert members? Questions for Dr. Caesar Ramthara? All right, if there are no questions, we'll move on to our next presenter, Gabriela Tandama from Mizoram University. Gabriel, are you available? Hello, Gabriel, are you available? I cannot unmute. Hold on. All right, just hold on. Oh, take. Ring tên na kan mo. Gabriel kalau unmute. Ro tira. Do we need to share your slide? Hello? Hello? We are unable to unmute from here. Please log out and log in again. Okay, so we'll while you prepare, we'll call the next presenter. Sorry, I'm back. Let me call Lansing Sani.
Can you hear me, Miss?
Yes?
It's me, Gabriel. Can you?
Okay, so can you present now?
Yes, I can present
All
it.
right.
Can you hear me?
Yes, yes, I can hear you loud and clear. All right, carry on then.
Okay. Is the slide visible, ma'am?
Yes, I can see your slide. Yes, carry on.
Okay. First of all, very good afternoon to the chairperson and then all the committee members and then the organizing committee and all the participants and the experts who are present today. I'll be presenting on the trend analysis of annual and seasonal relative humidity variability in Mizoram. And I'm a PhD research scholar under the Department of Geography and Natural Resource, Mizoram University. With the introduction, climate change and variability are among the biggest challenges of the world, and it somehow has a potential effect on Mizoram as well, as Mizoram is somewhat vulnerable to the effects of the climate change because our economy is mostly based on the agricultural activities and agricultural sector. And according to the IPCC 2007, the changes in the rainfall, temperature, and humidity trends and patterns are directly associated with the changing climate. Humidity, on the one hand, other than the other climatic variables, that is the rainfall and temperature, humidity, both the relative and the absolute humidity, is potentially a very insightful tool for studying climate. And water vapor, which is a part of the humidity, and the amount of the water which is present in the atmosphere as its transport around the atmosphere, is fundamental component of the hydrological cycle. The objectives of the study is to evaluate the change in the trend of relative humidity to have a better understanding of the potential impact of climate change in the state of Mizoram. The methodology which have been conducted in this study is the data which is used is for a time series period of 33 years, that is from 1986 to 2019. And it was collected from the Indian Meteorological Department, Science and Technology, Department of Science and Technology, ISO. And the technique which has been used is the non-parametric statistical tool man candle and sense slope test. The reason why I have used the man candle is because the climatic variables, the climatic data are non-parametric, and it doesn't have a defined pattern of data set. So non-parametric man candle is mostly used for climate change analysis. And then the data set has been categorized into two sets, that is the annual and then the seasonal group. The study area. The study area has been selected from the Northeast, that is Mizoram. And then the state has been divided into three sub-agroclimatic zones: humid mild tropical hill zones, humid subtropical hill zones, and humid temperate subalpine zone. The climate sub-agroclimatic zones, it's agriculture and climate has somewhat very strong relationship with each other. So based on the agricultural activities and the practices, the climatic zones has been divided into the four zones. And then regarding with the seasons in Mizoram has been divided into four by the meteorological department. That is the winter from November to Jan, spring from February to April, summer, that is May from August, and then the autumn is from September to October. So the results of the analysis, the man candle trend analysis and sense slope test shows that there is a positive trend in the humidity from 1986 to 2019, and then the annual average relative humidity, which is last in the table, that is from 1985 to 2019, it shows a 1.93 Z value. That is the normal cumulative distribution of the data set. And then within the table, you can see that the months of June, July, August, September, October, and November have a very high increase of positive trend of the humidity for last 33 years. And among these months, September has recorded the highest trend of humidity, increase of humidity. And these symbols represent the significance level. The significance level, that is 95% confidence, and then the 99% confidence level of p-value. And whereas among the seasons, the summer season and the autumn season has recorded to be the highest variability with the highest increase of humidity trend in the last 33 years. There's an overall increase, like we have just said, in the relative humidity. The year 2017 have been recorded with the highest range of humidity for a time period of 33 years, with 90.7% of annual relative humidity in 2017. And in 2017, there has been an increase of 16.13% of increase of relative humidity from its mean value. The month of August records the highest average of relative humidity for the period of 33 years with 89.76%. And in 2017, the month of September shows the highest increase of trend with an increase of 9.49% from its mean value. And whereas the autumn season has an increase of 11.03% mean value. And regarding with the discussions, and as we have learned that there is an increase of positive trends in the humidity. Humidity as a climatic variables plays an important role in determining the climatic system of a region. It somehow affects the climate conditions of a particular place. And then there is a direct relationship between humidity with agricultural system, the agricultural productions, human health, vector-borne diseases, and then the ecological system. In the earlier presentation also, there was a presentation based on the maize production with correlation the humidity and then the rainfall. In the results, you must have seen in the presentation that there is a direct relationship with the increase of humidity as well as the production of the maize. And with the vector-borne diseases, currently I'm working on one research, and then we have found out that with the increase of humidity in the state of Mizoram, there is an increase of malaria cases in the low elevated areas. And that it is necessary to have a scientific study on the pattern of the different climatic variables to understand the potential impact of climate change in order to formulate adaptive measures to combat the adverse effect of climate change in different sectors of human well-being as well as conserving natural environment. And this result somehow may help in a much better assessment of physical impact of vulnerability of climate change by policymakers and the government. Thank you very much.
Thank you very much, Gabriel, for your presentation. We now open the floor for questions from the experts. Any question for Gabriel? All right, if there are no questions, we'll move on to our next presenter. We will call Zairem Moi from Mizoram University. Zairem Moi.
Yes, yes.
Are you available? All right, all right. Do we need to share your slide?
No, Miss, I can share it from here.
All right, all right.
Đó là present now. Anh đưa trên gần làm tiếp theo.
Miss, can you please share the slide because I cannot share it from my computer.
All right, hold on. Are you able to see the slide on your screen?
Yes, Miss.
All right.
Okay, can I start? Okay.
You may start, yes.
Hello, everyone. My name is Zairem Moi, and I'm currently pursuing my PhD under Mizoram University. And today the topic of my presentation is on application of rock mass rating, a case study in Mizoram. Next slide, please. These are the contents of my presentation. Next slide. Our study focused on the study of rock mass rating to obtain the quality of rock along the state highway from Ngaidel to Tenzol. So, what is rock mass rating? Rock mass rating, which is commonly known as RMR, is a geomechanical classification of rocks used for obtaining the quality of a rock through RMR value. The RMR value can be calculated, and this involves 6 parameters such as the UCS, the RQD, the spacing discontinuity, the condition of discontinuity, groundwater condition, and orientation of discontinuity. And these parameters, based on the values, are giving ratings. And these ratings is based on the chart which was proposed by Binovsky in 1987. And then the sum of these ratings gives the RMR value. RMR is also used for designing rock slope and foundation, as well as for estimation of the in situ deformation modulus and strength of rock mass. Next slide, please. This is the chart which I've mentioned earlier, which was proposed by Binovsky. And as you can see, under each parameters, ratings are being given. And the different values of the parameters under there, we have the different values of ratings. Next slide, please. This is our study area, which lies along the state highway, and it runs from Ngaidel to Tenzol. And here are some, we have highlighted some sites of our study. Next slide. So our objective is to assess the quality of a rock in the selected site and to assess the vulnerability of the site, and also based on the result to suggest necessary action that must be taken. Next slide. So what type of method do we use to obtain the RMR value? As I've mentioned earlier, the 6 parameters, these values we have to obtain first. So for obtaining these values, field reconnaissance was done, and the UCS value was obtained with the help of locksmith rebound hammer method. Then from joint volume, we obtained the RQD value. Then from the thickness of the bed, the spacing discontinuity, and also, as you can see, the length, persistence, separation. From here, we obtain the condition of discontinuity. The attitudes of beds along with joints and slope, we obtain the orientation of discontinuity. And from the surface wetness of a rock, we can obtain the groundwater value. Next slide, please. I have given some of the results that we get from our study, and this is from Ngaizel. As you can see, the different spots, we have taken 10 different spots, and these different spots have different ratings, and these ratings, when they are added up, they give the RMR value from the second table. And as you can see, around 9 of the spots, they fall under the class number 5, which indicates that they have very poor rock. Next slide, please. Next slide, please. This is our study area, Ngaizel. Next slide. So from Ngaizel to Midumkham, another 4 spots are being taken, and these 4 spots, they have RMR value which are less than 20, and all of them fall under class number 5, indicating a very poor rock. Next slide. Again, in Midumkham, 10 spots were taken, and these spots were again analyzed with the help of RMR, and again 9 of the 10 spots selected, they fall under class number 5, indicating a very poor rock. Next slide. This is our study area, Midumkham. As you can see, the red lines indicate the part of our study area, Midumkham. Next slide. So from Midumkham to Thenzo, another 4 spots are being selected, and here also all the spots selected, they fall under class 5, indicating a very poor rock. Next slide. Next slide, please. Our study was solely conducted to attain quality of a rock along the state highway from Ngaizel to Thenzo with the help of RMR. And from our observation, the study site exhibited frequent rockfall throughout the year, mainly due to improper slope cutting, the slow tree, the weak lithology, and the frequent rainfall that occurs in our area. And the result from our study shows that more than 95% of the study area has a very low UCS and RQD value, indicating that they have a low strength, and also a moderately weathered to slight smooth surface, and the groundwater condition is mostly damp, and also the rocks are feasible and also consist of numerous joint sets. And from the study from the study conducted, we observed that most of the study have RMR which are less than 30. And according to Robertson 1988, if an RMR value is less than 30, then rockfall is inevitable. Therefore, in our study area, rockfall is inevitable, and the study area has high vulnerability to rockfall. So from our observation and results, action must be taken immediately and to prevent further occurrence of rockfall, and also to maybe, if possible, to prevent occurrence of rockfall. And this action must be taken under the guidance of a geologist and a civil engineer. That's all for my presentation. Thank you.
Thank you very much. We now open the time for questions from the expert.
So you have mentioned that some actions need to be taken. So what would you recommend being a geologist?
Actually, I'm still a research scholar. So I am not very confident, but I would definitely recommend the steel matching and other uh, rock anchoring. Can you hear me?
Okay.
Mm, mm.
All right. Are there any more questions for Zaire Moi? All right. If not, thank you, Zaire Moi, for your presentation. Um, uh, so I just want to take this time to introduce Professor Devesh Walia from the Department of Environmental Studies, NEHU, who has joined us as an expert member. Professor Walia, can you hear me? Professor Walia? I can see you.
Yes, you can see
All
me.
right, all
I'm
right.
there. Yeah,
All right.
yeah. Thank you.
Thank you, sir, for joining us. So the other expert members, I have already introduced them earlier, but let me introduce them again since you have just joined. So we have Dr. S.T. Dalzarzovi from the Department of Environmental Science, Mizoram University, Dr. Laldu Sanga Patwal, Department of Pharmacy, Assam University, and Dr. H. Lalruat Sanga, Department of Botany, Patsunga University College. All right, sir. Thank you. Now we will continue with our oral presentation.
Yeah, I've been listening to the presentation on malaria as well as this last presentation.
All
So,
right, all right.
yeah,
Thank you, thank you.
yeah.
All right, so if Lalming Sangi, if she is present, will call you for your presentation.
Okay, I'm present, ma'am.
All right, I can hear you. Do we need to share your slide?
Let me try. If I cannot, then I think I will need to share from your. Is it visible?
Yes, we can see your slide.
Okay.
All right, carry on.
Okay, thank you. A very good afternoon to you all. Myself, Lalvin Sangi, will give a presentation on the topic application of Total Station for landslide monitoring, a case study of Aizzul in Mizoram. And my teammates, Dr. Lalin Priya, Zagamuri, and H. Lalin Priya will be there to take up whatever questions you have raised. First of all, let's see what a total station is. A total station is an improvised version of modern surveying instrument. It is an instrument which is very useful and very commonly used for surveying and building construction by many geologists and civil engineers. We can define a total station as a combination of an electronic theodolites and an electronic distance meter. Because of this combination, it is possible to determine the coordinates of reflectors by aligning the instrument's crosshair on the reflectors and simultaneously measuring the vertical and horizontal angles and slope distance. As I have said, this total station can measure the data such as elevation, vertical and horizontal angle, and geotectic data. So it is possible to monitor the area which undergo movement. This measurement of horizontal angle, vertical angle, and the distance is easily done using this total station by analyzing the slope between itself, that means the total station, and a specific point. That specific point is what we call a target point, which is in the movement area. Here I'm going to highlight some of the methods which we use in monitoring the ground movement using this total station. First of all, we need to set up the instrument, and then we need to select the point, the station point, that is where we are going to place the instrument. And here, while selecting the station point, we have to make sure that all the target points, that is the points in the movement area, should be visible from this station point. And after the selection of this station point, we do a tripod setup and then mounting instrument on the tripod and focusing on surveying point and then leveling the instrument.
After doing different types of leveling, we do the orientation by backside, and I will not go into details all these things. And after orientation by backside, we need to select the targets, the points in that movement area. And after we select the target, we start doing monitoring regularly, and then we close the traverse. And here we have a diagram where we can explain more in details about the methods we use in monitoring ground movement using the total station. Here we have occupied point. This occupied point is a point where we place the instrument, the total station, and we have foresight and backside. This point are placed in an area which is undisturbed, which is stable. So these points are very important. By chance, if we miss our occupied point, we can trace back using the these two sites. And here we have a target 1, target 2, and target 3, and so on. We can make as many as target we want, depending upon the area of that moving area. And this target point, we measure the coordinates of all these target points from this total station, from this point, and whatever coordinates we measure, it is all recorded in this total station. Here we have an area in Zhuangtui. Here we did ground monitoring in different spots, and in this photo we show spots 2, 3, 4, 5 is not visible here. As I have mentioned before, this one is a total station. Here we place in a stable area which is undisturbed at all, and this point is a target point. This area is an area which face a movement, and we place a target point here. And this target point, the coordinates of these target points are measured from this total station. Here we can say that this is the margin between the movement area and the stable area. The stable area is this side where we place the total station, and here we mark the points where we took the coordinates. Like this, for 4 months, from starting from June, we did a monitoring of the ground movement, and whatever data we receive, we correlate them with the previous coordinates, previous data. So doing that, we get this type of graph. This is the here we plot the data where we took from spot 2, spot 3, spot 4, and spot 5 of target 1. And from this pattern, we can conclude, or we can interpret that total station is equipped with an electronic distance meter is widely used for landslide monitoring. And this total station employed to determine the rate of Zhuangtui subsidence movement. And the target fixed point at unstable sites are monitored from the occupied point, that is undisturbed site, and the observed values are correlated with the first reading. That means if we did the monitoring on August, then the data we get, it is correlated with the previous month data. And from that data, we observed that there is a slight movement in an inches in the Zhuangtui area. So from all this, we can say that a total station is a very easy and accurate monitoring device, but there is still errors at some points because of human. Thank you all.
Thank you very much, Ming Sangi, for your presentation. We will now open the time for questions from the experts. Are there any questions or comments? All right,
Yeah,
if
just
not,
ask one question.
Okay. Carry on.
Okay, has this been used before? Like this total station, has it been used to measure the landslide, or is this something new?
I think Bernardina can answer that one.
Okay, let me answer that question. So that's landslide forum monitoring of landslide monitoring that TS is very widely used, but in Mizoram it may be the first time. So in other areas, in European countries and in Indian also, that's widely used.
Okay, so this is the first use in Mizoram.
Pumadin. Hello, Sangte. Oh, is your total station runs to the used by the survey doing cadastral. The revenue department are also using this total station for land survey. Are they the same instrument? Sangte.
Hello. Hello, can you hear?
Survey
Hello, sir. Can you please repeat? My connection is not clear. I cannot hear your voice.
आधे घंटे का टाइम है कि ये प्रोग्राम चल रहा है, जब तक खत्म नहीं होगा ना तब तक उसके बाद चलेंगे। यार मेरी हालत खराब हो गई।
Sarwat Sang, can you please repeat the question for Ming Sangi? Hello? I think Sir is also facing some technical issue. All right. If there are no more questions or comments, we shall now thank Ming Sangi, and we will move on to our next presentation. The next presenter is Benjamin Changte from Mizoram University. Benjamin, are you available? Hello?
Yes, ma'am, I'm available.
All right, all right. Can you share your slide?
Yes, ma'am, I will share my slide. Is it visible, ma'am?
Not yet.
Is it visible, ma'am?
All right, yes.
Okay.
All right, you may start when you're ready.
Okay, ma'am. Thank you. Good afternoon, everyone. I would like to thank the chairman and the organizing committee for allowing me to present my topic of classification and provenance study of Buban sandstones around Kolasip district, Mizoram. First of all, let's take a look at the location of the study area from which the samples were taken. It lies in the coordinates of north 24 degrees, 14 minutes, 22.5 seconds, and east 92 degrees, 40 minutes, 53.7 seconds. And the samples were taken from Kualmoy Quarry, which is in Kolasip district. It is along the Kolasip road. And from the map here, you can also see the study area. Here are some field photographs. In figure number 2, you can see the outcrop within the study area, and in the next figure, which is figure number 3, you can even see calcareous nodules. Here is a little column of my study area and the points from which the samples were taken, and all the points are not depicted here. Only the interested samples from were depicted in this little column. So my objectives are to classify the Buban sandstone of Kolasip district and to decipher the provenance using petrographical studies. In all, we have taken around 15 samples, we have prepared thin sections for each of these samples, and out of which we have selected 9 samples for further studies. And these samples were analyzed using a Leica DFC 550 petrological microscope, which is fitted with high resolution camera, and the use of Leica image software. And acquired percentages of the minerals were then plotted using Microsoft spreadsheet, and the percentage which we acquired using the Microsoft spreadsheet were then plotted using Origin Pro 9.0 software. So here are the calculated percentages of quartz, feldspar, and the rock fragments using the SET software. From here you can see that we have 9 samples, sample number 1 to sample number 10, and from the table also you can see that we have total percentages of quartz, recalculated quartz, feldspar, and rock fragments which were used to plot the triangular diagrams. Here is a microscopic view of some of the minerals under XPL. From in the figure A, you can see microcline and king bands, and in figure B, you can even get a clearer picture of the king bands along with polycrystalline quartz and plagioclase. In figure C, you can see quartz inclusions, and in the last slide, we have a heavy mineral, which is identified as rutile. I'm sorry for the picture quality because the magnification, if we increase it, then the picture becomes a little bit blurred, so we have decreased the magnification to 10x only. So here is a classification of sandstone based on Folk 1980. These triangular diagrams are plotted using the Origin Pro 9.0 software. From here, we can see that the samples which we have analyzed comes under the lithic arenite. And based on Petitjean classification 1975, the same samples, most of them they fall under lithic arenite, while only some samples they fall under sublithic arenite area. So here we have the tectonic setting based on Dickinson et al. 1983, and from the samples and from the data which we have acquired, we can know that the sandstones, the tectonic settings of the sandstones belong to 2A, which is recycled origin. So here are my results. The framework grains encountered are mainly detrital in origin, with the most common being quartz, which constitutes between 60 and 70%. And next we have rock fragments, which constitute 19 to 31%. And lastly, we have a sparse occurrence of feldspars with only 3 to 10%. And we have observed some heavy minerals like garnet, rutile, to name a few, and these are found to constitute minor accessories. The encountered sandstones in the study area have been classified as lithic arenite, as the majority of the samples, they fall within the lithic arenite area as per classification based on Folk and Petitjean, while only a few samples, they fall within sublithic arenite according to Petitjean's classification. So my conclusion is that the Boban sandstones of the study area is classified as lithic arenite, and that based on different triangular plots, they reveal that the sandstones of the studied area, they show quartzolithic composition, low feldspar, very low volcanic grains, and abundant sedimentary as well as low-grade metamorphic lithic class. That indicates that the sands were derived from quartzose recycled orogeny province. Thank you.
Thank you very much, Benjamin, for your presentation. Now we open the floor for comments or questions from the expert members. Are there any questions for Benjamin? All right, if there are no questions for Benjamin, thank you. We will... Okay.
One small query from Benjamin. Just there is something. Yeah. So finally, Benjamin, you have found that the provenance of these Bhuvan sandstones, what is the provenance you said towards the end, just to end up?
Pardon, sir?
Yeah, provenance. Just can you show me your last slide once again?
Yes, sir.
Yeah. So provenance basically also means that from where you have got the material. Can you throw some more light on that? Provenance means the area from where the material has come.
Hello, sir.
Yeah, would you like to say something more about the provenance? You have just classified the Bhuvan sandstone. Classification is okay.
Yes, sir.
But the provenance of the sandstone, what would you like to say?
Yeah, sir, regarding that, it is mainly igneous and metamorphic reworked sediments, mainly from the Himalayas.
So what do you think? What may be the source? What is the age of these? And how from the Himalayas those things might have added there at Kolasep?
Because of marine transgression and regression. Sir, hello, sir. Because of marine transgression and regression.
So you think that from Himalayas these materials could have come?
Yes, sir. Exactly,
It's
sir.
okay. Yeah, we can go ahead. Thank you.
Yes.
Are there other questions for Benjamin? If not, thank you, Benjamin, for your presentation.
Thank you,
We
ma'am.
will now move on to our next presenter, Dr. C. Larim Rotfila. Dr. C. Larim Rotfila, are you available?
Yes, ma'am.
All right. All right. Please share your slide and continue with your presentation.
Hello, ma'am. Is my
Yes.
slide visible?
Yes, we can see your slide.
Okay, thank you. First of all, I would like to thank the chairman of this session and the organizers for giving us this wonderful opportunity to share our research on monitoring landslide activity of Bongkon, Aizawl using unmanned aerial vehicle. And this is a project funded by Directorate of Science and Technology, Government of Mizoram. The study area is a roadside section at Bongkon East, Lunglei Road, which is located at about north 23 degrees 45 minutes 4.320 seconds and east 92 degrees 44 minutes 2.400 seconds. The main aims and objectives of the study is to monitor the landslide area using unmanned aerial vehicle UAV and to prepare thematic layers of digital elevation model DEM, digital terrain model DTM, contour and 3D point cloud, and to compare the changes in morphology after two landslide events using hill shade aspect slope, contour and point cloud models, and to determine the causative factor of the landslide. Geologically, this area comprises of rocks of the Buban formations of the Surma group of rocks, which is exposing sandstone shale alternation of varying proportions with a few siltstones. And this section becomes increasingly sandy at the upper part of the section, showing a coarsening upward type of depositional environment and several sedimentary structures like lenticular bedding, heterolithic beddings and tight influence sedimentary structures are also found in this area. History of the landslide. Due to its steep topography, and also due to the construction of roads and anthropogenic activities like excavation for building construction, etc., this area has experienced several mass movements in the past few years. The landslide episode for our study area began with a natural landslide on August 2015. And due to recent excavation for building sites in the lower part of the section, it reoccurred on 15 June 2020 midnight. In this case, 20 to 30 families have to evacuate their homes due to fear, and several vehicles, including excavators, were buried. And due to heavy rainfall, it reactivated on 22nd September 2020, causing road blocks on both occasions. In this study, DJI Mavic 2 Pro drone is used to collect high-resolution images for the landslide. Using the onboard navigation system of DJI drone, which receives signals from GPS constellations. The field of view is 77 degrees, resolution of 1.4 centimeter pixel, with an accuracy of RMSE 0.7 meters. The flight plan was prepared using drone deploy flight application. The same flight plan was used to monitor the two landslide events. Flight altitude was set at 60 meters, and the overlap was set to 75% front lap and 70% side lap. A total of 136 images were captured. These captured images were all auto georeferenced through drone satellite integration using 15 to 18 satellites signals. And these images were used to create several thematic layers using drone deploy application. These are the thematic layers prepared at a scale of 1:300. First is the orthomosaic map, which gives a detailed, accurate photo representation of the area, which was created out of many overlapping georeference images that have been auto-rectified to increase the accuracy. These are the other thematic layers. The first row is the DEM, digital elevation model, which is the spatial extent of the elevation of the slide area. The second row gives the digital terrain model, which represents the background surface without any objects like plants and buildings. And the last row is the contour map, which were prepared from the DEM and DEM data. They were prepared at a scale of 1 meter to detect slight changes in the slight modifications in the slope, as seen in the change in the contour spacing in the last two images. The digital, using the DTM and DEM toolbox, hillshade, slope, and contour maps were also created and compared for the two landslide events using ArcGIS and QGIS software. This is a picture of the hillshade maps of the two landslide events, which is a grayscale representation of the landslide area with the sun's relative position taken into account, which shows the clear representation or the change in the topography of the slide area. Next is the aspect map. In this map, we identify the direction of the slope in our input data. In the first map, the large amount of dark blue colors in the crown area is northwest facing failure plains, which were removed after the second slide, as shown by the increase in the percentage of blue color, the light blue color, which is the southwest facing slope in the second landslide event. In slope map, each pixel shows the steepness of the area, and the higher values indicate higher slope. Decrease in the amount of the yellow color pixels in the second picture shows that the slope is much more stable after careful removal of the debris after the second landslide. Point cloud model or point cloud last files, which are generated from the drone data, are converted to grid files, which were compatible with surfer software. This file is used to generate the 3D model and the slope profile for the area and comparing the change in slope for the true landslide events. Geological data collection was done with the help of field move Clino application, particularly bedding joints and bedding and joints were collected for this study and its adjacent areas. The collected data were represented in this stereographic projection of planes and poles to planes. In the first image, the bedding planes are represented by the green lines and joints by yellow lines. The diagram shows the intersection of the bedding with the joints and also the presence of a large number of joint sets forming conjugate joint systems. This is a 3D model of the true landslide events, which shows two conjugate joint failure planes, J1 and J2, and these failure planes are still clearly visible after the second landslide. These are my results and discussions. Bongkon Lunglei Road landslide was monitored using UAV surveying. Monitoring dates were on 25th June and 11 November 2020. After the two landslide events, the affected area is 820.46 square meters calculated from drone deploy annotation software, and hillshade contour aspect slope maps were generated using GIS softwares, and they clearly indicate the change in the slope morphology. The beds strike between 320 degrees to 340 degrees and dip at about 30 degrees towards the northeast. And conjugate joint sets striking between 140 degrees to 180 degrees and 250 degrees to 350 degrees were observed. These are the joint sets which are the main factor for the failure in this area. As for my conclusions, the landslide was triggered by excavation work and conjugate joint systems supplemented by saturation, gravity, and other factors. Since the slide zone and its adjacent areas are still prone to mass failure, geotechnical investigation must precede any excavation and construction activities. UAV surveying can be used for monitoring landslide activities and also to monitoring engineering projects and to prepare progress reports, etc. Thank you.
Thank you very much, Dr. Sidarem Ratfela. Are there any questions from the expert members? Any questions for Rem Ratfela? So if not, thank you.
It's all right. It's okay. Yes.
All right, sir, continue. Carry on.
No, it's all right. It's all right. He has done extensive work, but he needs to be more organized while making presentation. That's what is one general comment.
Okay.
Because some of the things which were required in the beginning, he was speaking in towards the end. That's all. Rest of everything is okay. Thank you.
Okay, thank you. Yeah. So we'll move on to our next presenter, Dr. James Lalnunzira Shasel from Mizoram State Climate Change Cell. James. James, can you hear me? Hello, James. Okay, we'll share your slide from here. Dr. James, your slides are already up. Please start when you're ready. Hello. Dr. James is facing some technical issue. Just bear with us.
Hello.
Hello.
Yeah, hello. Hello, am I audible now?
No, we can't hear you. Please unmute.
Yeah, am I audible now?
All right. Yes, we can hear you.
Okay, okay.
You may start when you're ready.
Okay, thank you. Sorry for the inconveniences. Okay. First of all, I would like to thank the organizers and the esteemed experts and the participants. And good afternoon to all. Today, my topic is on rainfall-induced hazards analysis of Mizoram. And my name is James Lanuzira Rasel, and I am from Mizoram State Climate Change Cell, and I'm presenting this topic on behalf of my team. So, come to the objectives. The objectives is to analyze the correlation between hazards and rainfall in Mizoram. In the study area is the total geographical area of Mizoram, which comprises of 11 districts. And the data collection were done through State Meteorological Center, from which rainfall data from 1986 to 2019 were collected, and the hazards consisting of landslides, cyclones, hailstorms, floods, etc. were collected from disaster management and rehabilitation, which include 10 years record from 2009 to 2019. And the methodology adopted is for rainfall analysis, we adopted the rainfall anomaly index developed by ROF 1965, and which was further adapted by Freitas in 2005. And as you can see for the formula for yourselves, which have for positive anomalies and for negative anomalies. This rainfall anomaly index is used to classify the positive and negative severities of rainfall anomalies. From the precipitation data, the annual rainfall and the rainfall anomaly index can be used to analyze the frequency and intensity of dry and rainy years. And for statistical analysis, we use the polynomial quadratic regression model. And let's come. This table consists of the classification of rainfall anomaly index, which will classify whether those years were rainy or dry or neither neutral. And let's come to the results directly. And in case of rainfall anomaly index, during the study period, the rainfall anomaly index indicates that 2009 to 2011 years, the year with extreme rainfall,
with a score of 10.24 and 8.53, as you can see high in the graph, with excess of 1,761 millimeter during 2009 and 10, and with excess of 1,467 millimeter during 2010-11 from the average annual rainfall of 2,555 millimeter in the last 33 years. The analysis of the rainfall anomaly index reveals that there is a decline in the amount of the total annual rainfall since 2012 from the average annual rainfall of 1986 to 2019. It also reveals that there occurs an incidence of two extremely rainy years, that is 2010 to 2011, with 14 rainy years followed by 10 dry years and 7 very dry spells. In terms of landslides, the extreme rainy years, that is 2009-2011, resulted in 656 villages affected by landslide. During the monsoon season of 2010, Bazar locality of Mabit town were severely affected by subsidence and landslides, endangering the lives and properties of hundreds of people. About 50 families were evacuated and about 43 houses dismantled. This coincides with the year 2009 and 2010, when the received rainfall recorded the highest for the state during the past 3 decades. Within Aizawl city, around 10 numbers of houses were dismantled, which affected about 15 families and required evacuation during 2010 and 11. The following year, there was a long linear crack at Ramhun South Complex area. Around 10 number of houses were dismantled and almost 60 families were shifted to safe areas. The case of cyclone. The first incidence of cyclone was noticed in 1876 in Mizoram, and the year has been still remembered as Klichek Klekum, a local term for the year of cyclone. 1,100 villages affected by cyclone during 2009 to 2011. And in the case of floods, in June 2017, Mizoram faces the worst flooding of Klong River in 50 years. Hundreds were left homeless, compelling them to seek refuge in community halls and schools. This happened due to incessant rains amounting to almost 500 millimeters of rainfall just within the first 2 weeks of June. Approximately 350 houses have been completely submerged by the floods and 8 fatalities in the state. Many other areas have also reported bridges and roads collapsed, which has brought the traffic to a complete halt and cutting off interconnectivity of various district headquarters. Many water pipelines in the state have also been severely damaged and the pump house heavily sedimented. The flood also damaged 137 hectares of cultivated areas and 62 fish ponds. In Tlabung, which falls under Lunglei district in the southern part of Mizoram, more than 80 families have been evacuated from their homes and close to 70 houses have been submerged by the floods. Within Sermun, over 100 houses have been swept away by the floods. Let's come to the statistical analysis. In the case of landslide versus rainfall, the R-square shows a very high determination, and even in the case of cyclones versus rainfall, the R-square have very high value. And even for hailstorms, the R-square have very high of 73%, and with landslide versus rainfall anomaly index, we have the R-square of 0.80. And with the cyclones, in terms of rainfall and anomaly index, we have R-square of 0.88. And with hailstorms, in terms of rainfall and anomaly index, we have R-square equals to 0.72. So in the conclusion, the hazards occurrence and the number of villages being affected increases with heavy rainfall events. Anthropogenic and developmental activities such as slope cutting, filling, and disposing of sewage and drainage into slopes in a poorly controlled manner plays a very great role in triggering landslides. So proper implementation and enforcement of the Aizawl Municipal Corporation Site Development and Slope Modification Regulation 2017 is a very need of the hour, and the construction of proper drainage systems is required especially in the urban areas. Mock drills and awareness campaign to recuperate in case of occurrences of such incidences has to be taken by the concerned department. So these are some of the incidents damage caused during the 2017 incessant rainfall, rock slides, and connectivity, district interconnectivity is being destroyed, and the floods at Sairang village, and house damages by hailstorms, and the landslide at Bongkon, which my previous predecessor had discussed. So thank you.
Thank you very much, Dr. James Shasil. We will now open the time for questions from the expert members.
Yeah, Dr. James, it was a good presentation. And over the climate impact variability over the Mizoram region. I would like to know that you have done rainfall data. From how many stations you have collected the rainfall data?
8 stations, sir.
8 stations. So you think that 8 stations are okay to, they were chosen randomly or on the basis of some variability to represent the whole state?
Those 8 stations represent the 8 districts headquarters of the state. And I think that clearly represents the state of Mizoram.
So, on the basis of this, will you be able to prepare, say, 5 kilometer by 5 kilometer or 10 kilometer by 10 kilometer climate variability index or vulnerability index using this whatever data you have?
Yes, sir, we are working on that. We are working on that for, with IST Bangalore and IITM and with IIT Guwahati also. We are trying to make some climatic models like statistical downscaling model, all those things.
Yeah, you know that they have prepared such models, IIT
Yes,
Bangalore as well as not IITM, but some others, and it doesn't come practically true on the ground because the number of stations, as you have said, that 8 stations at for Meghalaya, it was for 10 stations. And so much recommendations they have given, and the government of Meghalaya through climate change center is in effect because nothing comes out to be true. So what do you think for Mizoram, how factual data it is on the basis of 8 stations?
So as far as concerned, these records are very consistent because they have been, it is manual. Manual collection, not the like of those, projected model.
Okay.
Yeah.
All right. So what do you think? Because the important link is that if you have got good data, you can definitely predict good model,
yes, sir.
workable, and you can say that that model will help in execution of some of the things which will be for the benefit of the people of Mizoram. So
Yes,
what
sir.
do you think that what are the still say some of the assumptions or some of the issues which you need to take care?
So, actually, we need to install more meteorological observation stations within the state. Like, for example, we have, moderate data for rainfall, but those are being recorded by the Department of Agriculture, but they do not record the temperature data. So, in order to address the climate change issue, I think we both need the temperature data and rainfall data simultaneously. So having only rainfall data at the moderate level and no temperature data very sparsely, so that will have a very biased, projection.
Yeah, that's what is the major issue. So good work, take care of all those things because actually the results should be executable. Then only, and if the people they get convinced, then only they try to adopt to whatever measures are being proposed by the government. So that's what in Meghalaya we have faced on the similar lines.
Yes, sir.
Because the people they don't believe the data, so they don't like to work as per the government recommendations. So there is a TRMM, that satellite imagery data is there of the rainfall. So at least after some period, you can get good data, and you can match the data with the ground data.
Yes, sir.
And then you can carry out good model. Good. Thank you very much.
Yes, sir. Thank you too.
Any more questions or comments? Or if not, thank you, Dr. James, for the very nice presentation. So we'll move on to the next presentation. Again, this is also James from Martin Luther Christian University, Thodam James Singh.
Yes, ma'am. Yes, ma'am.
Hello, James.
Yes, ma'am.
Hello, James.
Yes, sir.
We can hear you.
Okay. So I'll just share my slide.
Okay.
Yeah, just a second.
Just give us a moment. We are unable to see your slide.
Yeah. Yeah, it's just buffering, I think. Presenting.
Do you want us to share the slide from here?
Yes, ma'am. I'm just trying actually. Yeah. It would be good to do from this side. Just 1 second, ma'am. I think, ma'am, I'm getting some technical problem out here.
No problem. We'll share from here.
Sure, ma'am. Sure, ma'am. Yeah.
All right. Can you see your slide?
Yes. Thank you so much,
All right.
ma'am.
You may start,
First of all, sure. First of all, thank you, team of Mizoram Science Congress. A very good afternoon to the respected experts, teachers, and their friends. The title of my topic is vegetational changes in the eco-sensitive zone of Nongkilam Wildlife Sanctuary, Riboi district in Meghalaya. Species diversity, as everybody knows, is an important attribute of a natural community which influences the functioning of an ecosystem, stability of the community. Due to the repeated erosion of the biodiversity, there are reduction in the number of species, dispersal and migration processes, ecosystem inputs and outputs. These are some of the mechanisms responsible for the erosion of the biodiversity. According to World Bank, Northeast India might be facing inescapable rate of deforestation due to dependency of the people in the forest area. And 40 to 60% of the income of the people living around the forest got from the forest of the adjacent habitat area. Next slide. Yeah, about 40% of the poor country are living in the forest fringes. And like anthropogenic activities, this leads to the fragmentation, landscape alteration, degradation of the wildlife, and the practice of the monoculture, tree plantations, and these are some of the major threats to the survival of wildlife species, especially Schedule 1 species. Next, please. Yeah, Like Northeast comes under Indo-biodiversity hotspot, and it contributes 23.94 in the India's forest cover. And Meghalaya have 73.66% of geographical area under forest cover, and the 5% of its geographical area is under the state control, and the remaining comes under district autonomous district council, community and private land. Nongkilam Wildlife Sanctuary again comes under this biodiversity of hotspot, and many authors has reported call for conservation of and protection of this area. J.J. Joseph reported 1036 taxa of vegetations and many other important species in this area. Even he further stated that a unit of the South Indian forest of Western Ghat, another biodiversity hotspot, harbor less number of species as compared to this forest. Next, please. Yeah, some major problems like shifting cultivation, exploitation of the wood, and the basic requirements of the villages. These are some of the environmental threats facing by this sanctuary, and many mammals and endangered species has been reported by many eminent authors. Working plan of the Nongkilam Wildlife Sanctuary. Next, please. Yeah. Yeah, even the state government of Meghalaya has reported about the clearing of the timber and the firewood, as well as the agriculture plantation around this sanctuary in the eco-sensitive zone. And the most important problem is the dependency of the fuelwood in the area. Yeah, now this is a definition of sanctuary as per the Wildlife Protection Act 1972, which is of an adequate ecological, faunal, floral, geological, morphological, natural or geological significance. And the state can define the sanctuary under Section 18 of the Wildlife Protection Act. The objective is like to identify the type of the vegetation and nature of the changes undergoing in the protected forest and the change and the villages in the EAZ of NWLS, and to suggest the appropriate measures of the improvement in this area. The study area covers around 202 square kilometers, which includes 125 of the government land and 74 square kilometers of the district council and community land. Altitude is 200 meters to 965. ESZ extent is from 335 meters to 8.43 kilometers. Important rivers like Umran is there. This is a study area map, which shows the yellow stripe is the wildlife sanctuary. Please come to the next slide, please. Yeah. Yeah, yellow stripe shows the wildlife sanctuary, and the dotted black boundary is the boundary of the eco-sensitive zone of this area. Next, please. So these are some of the pictures of the important rivers around this sanctuary, which is bordering the sanctuary and the village area. Next, please. Yeah, the research design, it involved a quantitative approach, and a sample unit. We have taken the Nongkilam Reserve Forest, Wildlife Sanctuary, and the surrounding 4 villages, Lailat, Umladu, Umtasor, Nongkinri. So sampling method is like vegetation analysis following the Mishra and the Muller Dombios and the Ellenberg was carried out on a seasonal basis. And the quadrant of 10 square meter and 5 meter square and 1 meter squares are used for trees, shrubs, and the herbs. So IVI calculations, then Shannon Diversity Index, Sorensen Similarity Index has been applied. And the book of Nongpo, and Flora of Nongpo by J.J. Joseph and J. Joseph, and has been used for identification of the plant. These are some formulas which I have applied in calculating the diversity, similarity, and IVI. Yeah, please come to the next. Yeah, next. Oh, some of the photographs. These are some photographs. Next, please. Yeah, some of the photographs of the sample plot taken in the NWLS, Nongkilam Wildlife Sanctuary. Next, please. Just keep on. These are some photographs which I have taken in the village area. Yeah. These are some photographs showing the collection of the beetle leaf and the ready for transportation. Again, in the village area of the Laila village, Umlado village. Yeah. Please go on. Yeah. Yes, just a minute there. Yeah. Next, please back. Back, please. Yeah, well, just a minute here. Actually, this is the area they have burned down just to do for cultivation. This is just adjacent to the sanctuary area. So most of the practices used here is like plantation of the kwai, banana, and the beetle leaves, etc. So these are some of the activities taken by the villages. Next, please. Next, please. Yeah, some calculation of the IBI of the area adjacent to the villages. Yeah, next, please. Of the 4 villages like Umtasor. Go on, please. Yeah, Umlado, then go on, please. Yeah, next, Lailat. Yeah, next, Nongkinri. So these are some IVI calculation. Come to the next, please. Abstract. Yeah, just about. Yeah. So this is the IVI calculation of each villages adjacent to the forest. So what I found is like the 1 minute there. Yeah, so Sorea robusta, Sima Wallace, these are some of the species with the highest IVI value. Even the Senen diversity index shows the maximum in the Lailat village and the minimum in the Nongkinri village. Please go on, please. Yeah, this is a similarity index showing the Umtasal got the highest similarity index compared to the other villages and the minimum in the Umlado. Yeah, go on, please. Yeah, these are some of the forest type which I found in the area. Go on, please. Yeah, coming to the result and discussion. Now, in my yeah, back please. Yeah, just back. Yeah. So in the forest area, total number of the species which I recorded is 331 numbers and 239 numbers in the village area. And the IVI species ranges from 26% to 64 ranges with the species like Sorea robusta, Sima, Tectona, Trivia notiflora, Caria arborea, etc. And the maximum species diversity is found in the Lailat village, 2.92, and the minimum is found in Nongkinri village. So, for instance, similarity indexes was maximum in the Umtasor and minimum in the Umlado village. So, the most important thing which I recovered is that the reduced flora diversity is maximum in the forest area, comparing with the in the forest area, comparing with the protected forest, and the plantation of broom, betel nut, banana, rubbers has fragmented the forest area in the village region. Next, please. Yeah, secondary growth of the bamboo, then macaranga, then the like weedicides, eupatorium, even lantana are expanding in the area because of the disturbance of the anthropogenic pressure. Then forest producers extractions. I mean, forest producers extraction are minimal as the forest area is all disturbed by the cultivation of plantation of these other cash crops and all. So the remaining patches which I found in the village area are of like very small girth. They are of the pole size comparing to the protected area. And the natural regeneration, of course, is very good in the area with the luxuriant growth of the herbs and the shrub after the rainy season. However, the proper management and the maintenance is required. Yeah. Now, coming to the conclusion, it is very important to ensure the proper enforcement of the ESZ rules and to promote the regulated ecotourism and to provide the conservation friendly sustainable livelihood earnings and to preserve the pristine condition of the area, which would be a source of the livelihood of the villages. A plantation of the fast growing tree species may be one of the important things to be applied in this area. And scientific and technical approach of the agriculture purposes are one of the important factors which need to be applied here. Social awareness on environmental issues, establishment of the higher education institutions, well-equipped hospitals are required, but not the least, providing alternate source of income by providing skilled job training is must. And coming to the next, please. I would like to end my slide by Chinese proverb, by using this Chinese proverb. You give a poor man a fish and you feed him for a day. You teach him to fish and you give him an occupation that will feed him for a lifetime. Thank you so much.
Thank you very much, James Singh, for your very nice presentation. Now the floor is open for questions from the expert members. Are there any questions for James Singh?
Yeah, thank you, James. It was very extensive work you have carried out.
Thank you, sir. Good afternoon, sir.
Almost everything that became very lengthy. But it's
So,
okay.
sir, thank you.
Keep on doing good.
Sir,
Thank you.
sir, actually, it's an ongoing project, sir. I have to still go on for some more.
Yeah, it's all right. It's okay. Yeah,
thank you, sir. Thank you, sir.
All right, thank you. We'll move on to our next presentation. We'll call Ashim Rahul Singh for a presentation. He is from the Directorate of Environment and Climate Change, Manipur. Hello, Ashim Rahul. Please unmute your mic. Please unmute.
Hello.
Hello, yes, we
Hello.
can hear you.
Good afternoon, madam. Can you
Good
please share
afternoon.
the slide from your side?
Yes.
The net is working properly here in my room, so
No problem. We'll share it from here. Are you able to see the slide on your screen?
yeah.
All right, you may start when you're ready.
Good afternoon to all. First of all, let's fight the coronavirus. Be brave, strong, and please surely don't shake hands. I hope the coming year will surely bring a new life to all of us. By the way, my name is Asim Raul Singh. I'm working as a project scientist in State Climate Change Cell, Manipur. And here goes my topic. The title of my topic is Climate Variability and Change, a study on Manipur floods. With the help of my colleague, I complete this presentation. Before coming to my slide, as a brief introduction, let me share about my state, the Manipur. The state is like an outlying area of rugged hills and narrow valleys, inner area of floodplain. The state occupies a geographical areas of 22,327 square kilometers, which is 0.7%. Valley cover only 10% of the total geographical forest. Average elevation is about 790 meters above the mean sea level, and depth of the hill is about 1,500 meters to 1,800 meters. Next slide, please. Next. Thank you. Well, flood disruption is an issue to discuss. The state witnessed severe flood disruption almost annually at this decades. The highest rainfall intensity occurred during the monsoon season, followed by pre-monsoon, post-monsoon, and then the winter season. In fact, the change in the rainfall patterns and increase in precipitation is a clear indication of the changing monsoon in Manipur. Many of us believe the sign as a result of climate variable and change. To understand the knowledge on climate and climate adaptation plan is one way forward option in reducing the hazards. Here, the slide represents the weather and major rivers of Manipur, which is held responsible for the worst scenario of flood situation in a state. Next slide, please, ma'am. Well, I have given only one objective. By the way, I have given only one objective, only to make sure that the climate is responsible for the occurrence of flood in the state. It is also reported in the IPCC 2007 that extreme wet climate are projected to become more severe in many areas, whereas mean precipitation is expected to increase and dries. Extreme are projected to become more severe in areas where mean precipitation is projected to decrease. As a result, more flooding will be seen in most tropical regions like Manipur and Asian monsoon regions. So here is my objective: to obtain a clear idea about the scenario of climate change on the extreme weather event like flood in the state like Manipur's. Next slide, please, ma'am. For the methods and methodology, it's depend on the availability of data, at least 20 data of continuous and complete observation until 2018. Study the full season clearly. Daily data were considered initially. Weather stations were kept at 15 different locations of the state to understand the weather events. Next slide, please, ma'am. The result, according to my calculation, I divide the result into three parts: main flood areas, flood frequency, and flood seasonality. For the main flood area, with the help of secondary data, I depend on Manipur Disaster Management Department records, and scientific review to define the worst scenario of flood years. The following are some details of worst scenario of flood in the state. Next is next slide, please. Flood for the flood frequency, I have given only 3 scenarios. That is one of the worst scenario of flood event in Manipur. Here, the slide represents those 3 points that I like to be considered. Next slide. And the last one is flood seasonality. For this, I studied the pre-monsoon season, post-monsoon season, and winter plus monsoon season. Next slide, please. Finally, this is the last part of my presentation. That is the concluding part. Here I read some of the need of time to tackle the problem of flood scenario in Manipur. I think concerned department, NGO, and other activists, if possible, can activate such activities now during the time of flood, or with the help of these some points given in the slides, we can easily maintain and strengthen the riverbanks, and defending the riverbed is necessary if possible. Improving the drainage facilities in urban areas, check dams, and other environmental related efforts to regulate the water flows, or like restoring traditional recharging structure. This one is the most important, I think, in our state, and water bodies are important in controlling the sudden floods. Local communities should be equipped with climate change knowledge. And finally, the last one is extension of climate technology to offset the vulnerabilities with effective climate adaptation plan. And next part. These are some of the flood calendars happening in Manipur. Next. Please next. Next, ma'am. These are some photos that I collected during that events of flood in 2017. Next. Thank you.
Thank you very much, Rahul Singh, for your presentation. Now we will open the floor for comments, questions, or questions from the expert members.
Okay.
Yeah, Mr. Singh, you are trying to link the floods with the climate change, isn't it?
Yes. Yes, sir.
So will you tell me that how much temperature or variability is there recorded in the past 100 years in your area, in Manipur area?
Before that, it was 15 to 20, and after that, after this coming decades, it suddenly come to higher, like 30 to 35 in this decade, sir.
Because in Meghalaya, we have carried out the temperature change in last 100 years is around 0.5 degrees centigrade. So that also brings in lot of variability.
Yes,
but
sir.
Can you directly link it with these floods with climate change?
Not really, sir. Just trying
to link them.
Just trying to correlate it with this with the flood, sir.
All right. So accordingly, you need to propose some measures to be taken so that the floods, the river water that is retained in the river channel, isn't it? That will help
Yes,
taking up the studies. All right, it's okay. You are trying your best to carry out good.
Thank you, sir.
Okay.
All right, if there are no more questions, we'll call our next presenter, Abanda Palay Walang from Martin Luther Christian University. Abanda Palay, can you hear me?
I'm just, hello?
Oh,
Can
hello,
you hear
yes.
me?
Yes, we can hear you.
All right, I'm just sharing my screen.
If you have any technical problem, we can help you from here.
I'm just I just have to click share, right?
I think so.
Because
Is your slide,
let me
is
try
your
again.
is your PowerPoint open in your
Yeah, it is
laptop?
open. Yeah, it
Yes.
is open.
Yeah,
So,
technically you're just supposed to click on share presentation.
yeah, I'll just click on this. Yeah.
Do you
I
see
think I
the
got it. I
present
got it.
present now? Yes.
Yeah, I think it's
All right, it's coming up.
it's visible there, right?
Yes, yes, we can see your slides.
All right. Okay.
All right, you may start when you're ready.
Yeah, thank you very much. A very good afternoon, everyone, respected expert members and fellow participants. My name is Abanda Paliwalang from the Department of Environment and Traditional Ecosystems, Martin Luther Christian University, Shillong. I'm presenting a joint paper on the role of traditional ecological knowledge in community forest conservation, Meghalaya. Traditional ecological knowledge or indigenous ecological knowledge, often used interchangeably, refers to knowledge about natural resources, utilization, and conservation of the same. As defined by Burks, it is a cumulative body of knowledge, practice, and belief, evolving by adaptive processes and handed down through generations by cultural transmission about the relationship of living things, including humans, with one another and with the environment. TEK finds its significant application in biological and ecological studies, conservation, resource management, pharmaceutical, and preservation of culture itself. Among the indigenous communities of Meghalaya, the Khasi, Jaintia, and Garo tribes, traditional knowledge is used in forest management, water conservation, ethnobotany, agriculture, irrigation, hunting, fishing, etc. In the East Khasi Hills district of Meghalaya, community forests are classified into group of village forests, privately owned forests, clan forests, sacred forests, village protected forests. Community forests often provide food, raw materials, medicinal resources, regulate soil nutrients, air quality, pollination, maintain species, and genetic diversity. In Meghalaya, two-thirds of its ecoregion is either lost or degraded. 25% of the forests in Meghalaya are highly vulnerable to climate change. A review by Karkongor and Tiwari suggested that most of the community forests in Meghalaya were either degraded and commercially exploited. Sacred forests in Swe, as recorded by Mishra et al. 2004, suggested that the forests are fragmented and degraded due to grazing, felling of trees, and collection of firewood. Although at present the village community has prohibited logging and extraction of firewood, grazing is still allowed in both the sacred and village protected forests. The present study highlights the efficacy of traditional knowledge in forest conservation and the current status of forest diversity in Swe. The objectives are to identify and assess the role of traditional ecological knowledge in community forest conservation, to ascertain the current level of biodiversity in both the sacred forest and village protected forests, to establish a link between traditional ecological knowledge and community forest conservation. Area of research study. Swer is located in the East Khasi Hill district of Meghalaya at an latitude of 91 degree 47 minute east and longitude 25 degree 25 minute north, and an altitude of 1915 meter above sea level, covering an area of 1624.72 hectare. The village has a total population of about 1,365 people with 386 households. Sacred Forest comprises an area of about 13.2 hectare and the village protected forest an area about 18.9 hectare. The entire village community still practices the traditional religion. The community forest is owned and managed by the village community itself, inhabited by several clans, including the Sinrem, Nongse, etc., with the clan Swer as the principal clan. The forest is under the supervision of the Basan Swer, who is also the elder of the Swer clan. Research design. A mixed method study was incorporated in this study using both quantitative and qualitative methods. Non-probability semi-structured interview was administered to get a knowledge on the type of traditional knowledge practice, the utilization of forests and forest products, and the dependence on ecosystem services. A vegetational analysis following Misra and Mueller was carried out in the year 2019-2020 using quadrants 10 meters square, 5 meters square, and 1 meter square for trees, shrubs, and herbs respectively. Collected plant samples have been identified with the flora of Assam, flora of Johor, and the herbarium at the Botanical Survey of India. Soil samples were collected from a depth of 0 to 15 centimeter from both the sacred forest and village protected forest. Analysis of water for its potability from the spring water shed was also carried out for the parameters according to the Indian standard for drinking water. Results. Through the interview with the locals, various types of ecosystem services have been listed in Table 1, which include water, cultivated land, grazing, NTFPs, timber for the ongoing road construction, religious rites and rituals. The NTFPs are collected at various times of the year by the local inhabitants as listed in Table 2, firewood, forest litter, fodder for livestock, fruits, mushroom, medicinal herbs. These are the pictures of the local women gathering mushroom from the sacred forest, and on the right hand side are the women collecting firewood from the village forest. Floristic composition. The vegetational sampling carried out revealed the presence of 68 numbers of identified plant species, including 18 tree species, 14 shrubs, and 9 climbers. Among the monocots, the Poaceae showed greater distribution in the disturbed area of the sacred forest. About 13 epiphytic plant species were recorded, including ornamental orchids like Dendrobium, Batoculus, Iria melaxus, etc. The study also recorded the presence of 7 medicinal plants, 12 ornamental, 1 endemic, 4 edible, 1 endangered, 2 rare, and 1 insectivorous plant species. The IVI were recorded for Rhododendron arboreum, Myrica esculenta, and Symplochus glomerata, were the highest at 63.0, 40.46, and 29.06 respectively. These are the list of the plant species that have been identified, even though there are several species that haven't identified yet, but will be done in the future. These are the list of the IVI of the most common tree species in both the sample sites. Soil quality analysis showed that the soil is acidic in nature with a higher concentration of our organic carbon and available nitrogen. Accumulation of large amount of litter is also evident in both the sample sites. Water quality of the spring water source showed some presence of total coliform, which might be due to the presence of a sanitary facility nearby the source. Retesting of the water will also be followed in the future. Discussions. Traditional methods of conservation of community forests. The community participation in conservation generally extends beyond the community forest as depicted in Table 7, type of conservation methods, and the traditional ecological knowledge used. Forest, classifying of forests into sacred, village protected forests, and village forests based on the utilization and conservation purposes. Spring water, preserving of a surrounded hillock to replenish the source. Rainwater, harvesting of rainwater to replenish the forest and to meet the needs of the community. Construction of fire lines. Fire lines about 20 feet are made annually around the village forest by the community people to prevent and control the spread of forest fires towards the sacred and village protected forest. The increased number of plant species, including the proliferation in the number of epiphytes, fungi, and lichens reflect the positive outcome of such initiatives. Plantation of trees inside the community forest for future use is no longer permitted like before. Though the process of identification of plant species recorded so far is still undergoing, the available data point towards an increase in plant diversity as compared to the study carried out by Mishra et al. In 2004. Enrichment in soil nutrient reflects a tendency towards recovering. Water conservation measures in the vicinity of the community forest have helped in forest rejuvenation and availability of drinking water for the local residents. Sorry. These are the images of the traditional spring water conservation and management. On the right hand side is the traditional rainwater harvesting. Threats to conservation. Over extraction of NTFPs may eventually lead to forest degradation if not checked. Hunting of migratory birds during winter may affect dispersal of seeds. Small fire patches often seen in the forest may lead to forest fire and facilitate species loss. Introduction of invasive species like Pinus petula in the protected forest may harm the ecosystem balance. Cultivation at the foothills may also cause land degradation. Lack of avenues for livelihood may increase the pressure on the forest and its resources in the future. Conclusion. As it is evident from the study, traditional ecological knowledge has played a significant role in conserving the Swer community forest. Traditional ecological knowledge is handed down from the past orally through generations, and at present there are around 3 to 4 village elders who are still the custodians of the traditional ecological knowledge. Classifying of community forests in terms of utilization aids in conservation and enhances in imparting diversity and richness to the forest ecosystem. Measures like prohibition of tree felling, collection of firewood, cultivation and plantation of trees in the protected forest have been followed by the whole community. Such measures have paved the way for forests to gradually recover from the process of degradation, rejuvenate the forests and water bodies, and maintain the ecological balance. However, overutilization of NTFPs, cultivation at the foothills, bird hunting, introduction of invasive species may have a negative impact on the forest ecosystem. Awareness on conserving and judicious use of NTFPs may go a long way in preserving such biodiverse forests. These are the references used. Thank you very much.
Thank you very much, Abanda Walang, for the presentation.
Thank you.
Now the floor is open for any comments or questions from the experts.
Yeah, I would like to get some recommendation and suggestion. That will be very helpful.
It's good presentation.
Thank you, sir.
I think the presentation is well received.
But
So
only
thank you.
so much.
observation is that it became very lengthy also.
Mm-hmm.
Yeah.
Okay.
Thank
Sorry,
you.
hello.
Mm-hmm. We can hear you.
Yeah, I just wanted to say that you have made it very lengthy. That's what. Rest everything is okay. Because some of the in the process now, some of the things were not linking to the work. Basically, you want to represent or you want to present.
Okay, I'll keep that in mind.
It's all right. Otherwise, it is okay.
Thank you. Thank you, sir.
All right. Thank you, Abanda.
Thank you very much.
Now we'll move on to our next presenter, Silal Mwankimi from Government Zirtiri Residential Science College. Silal Mwankimi, are you available?
Yes, ma'am. Yes. Hello. I'll do my
Hello.
presentation. My side from here. Okay. Yes.
All
hello.
right.
I'm here.
Yes,
Hello?
we can hear you. We can hear you.
Okay. I'm sorry. Okay. Hello. Okay. Let me see.
Will you be sharing the slide from your end?
I will share from here. Okay, I'll try. Okay.
All right.
I'm trying. Just 1 second. Okay.
So while Montimi is getting ready, after this we have only one more
Okay.
presentation from the art sciences, but we have left behind two presentations from the life sciences. So after this art sciences, we will move back to life science for just two presentations. So in total, we still have 4 presentations to go. I know it has been a very lengthy session today. And I thank all of you for your patience and for bearing with us till the afternoon. So kindly bear with us for just 4 more presentations. Hello, yes, we can hear you.
Yes. Can you see it, ma'am? Hello?
I can't see your
Hello?
slide. Hello, I can hear you, but I can't see your slide.
Okay.
Shall we
Okay, can
try
you
sharing?
please-
All right, I think it's coming up.
I'm
Yes.
starting. Okay. Okay.
Okay.
Okay.
All right, you may start.
Okay. Thank you. Okay. So my topic is geochemical study on Buban shells in New Jeans section of Aizawl district, Mizoram, India. My name is Dr. Silan Mankimi, Department of Geology, Government City Residential Science College. And this is the first time ever reported on geochemical study on Buban shells from Mizoram. Mizoram is an accretion sedimentary basin of Miocene age related to Himalayan orogeny. In this paper, we focus on Buban shell in the vicinity of Muti village, which belongs to Upper Buban unit of Buban Formation, Surma group. So it is lower to middle Miocene age. I think I don't have to read all these things. So locations and geology of the study area. This is the study area is in the northern part of Aizawl district. In India topo sheet, it is 84A/13 and it is located nearly 15 kilometers northeast of Aizawl city in Mizoram. The rock succession in the vicinity of Muti village constitutes a part of the Upper Buban unit of the Buban Formation. As I have already mentioned it, so I think I have to skip this one. And this is my location map. Here you can see that Muti is in the northeast part of Aizawl district. This is Aizawl and this is Muti village. So I collected the samples from here. Materials and methods. So infield shells are gray to dark gray and brownish in color. They are thinly laminated, crumple, and are splintery in nature. Samples from the exposed shells were collected for geochemical study. So I wash the collected samples with distilled water in order to prevent contamination, and I prepare it for analytical work. I did my analytical work at CSIR, National Geophysical Research Institute, Hyderabad. So I did 4 major oxides in X-ray fluorescence, trace and re-elements in ICP-MS inductively coupled mass spectrometry. for trace and re. And I put together results and discussion since we don't have our time is limit. So this is the result. So I think I also have to skip this one. I'll show this this result. This is the concentrations of major oxides in weight percentage and sum of the elemental ratio in the Buban shells. The concentrations of SiO2 is high, varies from 62 to 85 to 73.22 percentage weight, but Al2O3 shows a small variation. You can see that Al2O3 here, and the elemental ratio of Al2 DiO2 varies from 18.05 to 21.91 with average value of 19.63. So I'll go to the result of trace element. This is the result of trace element. Trace element was normalized with post-Archean American shell. So I did normalize with past 1985. In Buban shells, SR strontium are enriched, which is 49.37 to 143.82 ppm per parts million. The enrichment of strontium may be due to the accessions of calcite minerals in the Buban shells. And also the content of barium varies from 2. Let me show you barium here. This is the concentration of Buban shells. So it varies from 2.993.1 to 714.49 per parts million. Barium enrichment in sedimentary rocks can be considered as an indicator of detrital flux. Rare earth elements. In Buban shells, the rare earth element shows a large variation, 145.8 to 303.48 ppm. And the shells shows light re-enriched and flat heavy rare earth element with negative anomaly. The elemental ratios of rare earth elements in the shells are shown, and also observe that the shells show less fractionations of rhenium, lanthanum, and terbium. It varies from 6.78 to 14.48. And then it is between the post-American shell. This is the result of rare earth elements. Here you can see that rare earth elements in Buban shell. So I did normalize with Byton 1984. So you can see that light re, the enrichment of light re and flat pattern of heavy mineral, heavy rare earth elements. For geochemical classifications, I follow Heron 1988 classification diagram. In Buban shell, the chemical compositions is classified as WEC and lithorenite. WEC is a dirty sandstone which composed of pyroxene, amphiboles, feldspar cores, and lithurnite is rock fragments, which is more than feldspar grains. Paleoweathering. In Buban shells, the intensity, the rate of weathering is very high, and most of the sample falls into elite. Elite is the minerals of clay. And tectonic setting. So in the Buban shell, the tectonic setting diagram suggests that active continental margin here in provenance, okay? As we all know that the tectonic setting is closely relationship with provenance. The average of K2O3, Al2O3 varies from 0.15 to 0.22 with the average value of 0.18. As I have already shown that the result of major oxides in the previous slide, including the elemental ratio. So the elemental ratios are closely related to the upper limit of the clay mineral range, which indicated that the elite is the dominant clay mineral in the Buban shells. In this study, the thorium, scandium, thorium, cobalt, thorium, chromium, and thorium and lanthanum, scandium ratios are compared with those of sediments derived from felsic and basic rocks, as well as to upper continental crust, UCC, and post-American, post-Archean American shells. So here also you can see that the Buban shells in Mizoram shows the mudstone, sandstone, and basic and acidic. The concentrations are very complex here. So in this study, we conclude that the Buban shells was deposited in active continental margin with high intensity of chemical weathering. And these shells is classified as WEC and lithurnite. So the source rock of the sediments were complex that they were highly depleted and felsic source. Elite is the most dominant clay mineral in the Buban shells in Mizoram. Thank you.
Thank you, Dr. Silal Mwankimi, for your presentation. So now the floor is open for comments or questions from experts.
MK.
Yeah.
Ruat Sanga speaking.
Yes.
Okay. It's, I think, a good composer. And you might think my question being asked is very silly, but you know, what is the composition richness or diversity indicating? It enhances, it adds to the stability of the rock, or it it enriches the nearby soils. What's the significance of knowing all those compositions and the richness of elements in a particular rock body?
Okay. Thank you for your question, sir. You know that I collected the sample from the upper Buban shells only. So in this study, I have found out that the clay minerals. Most of the clay minerals of illite are the most dominant minerals in upper Boban shales. So it means that it is more stable than upper shales.
Okay. Has something to do with the fertility of the adjoining soil as a result of weathering and leaching of the rock.
It's supposed to be more stable than other shales if the dominant mineral is illite.
Okay, thank you.
All right. There are no further questions. Next presenter, Lalong Kimi. We'll request Lalong Kimi to present her poster. Lalong Kimi, can you hear me? Are you available? Lomkimi may be facing some technical issue, so we'll be sharing the poster from here. Wankimi, please, can you leave the presentation? Can you leave? Can you end your presentation?
Okay, sorry. Oh, okay.
So Lomkimi was available earlier, but she is facing some technical problems. So anyway, since this is a poster presentation and doesn't necessarily require the live presentation by the author, so we'll just show it for 1-2 minutes, and then we'll go to the next presenter. So this is a non-timber forest products of Troy Wildlife Sanctuary. So as I have mentioned before, we left two presenters from the life sciences section. So we'll quickly go back and I will request Charles Lalnunfela from Mizoram University to be ready with his presentation. Charles, are you there? Can you hear me?
Yes. Can you hear me?
All right. Yes, I can hear you loud and clear. All right. So do you want us to share the slide from here?
Yeah, just share it from your end.
All right. Can you see the slides?
Not yet. Wait.
It's already up.
I can see it. Thank you.
All right. You may start
Yes,
when you're ready.
All right. Yeah. Greetings to the session chairperson, expert members, and all the participants. It is a great privilege that my work entitled Unfurling the Treasury of Ilex Cassiana and Quantification of Quercetin using HPTLC method has been selected for oral presentation at Mizoram Science Congress 2020. As mentioned, Charles Lalnunfela from the Department of Mizoram University, and my co-hosts are listed in the slide. Okay, next slide. At the outset, I would like to stress on the selected plant that is Ilex Cassiana, because not much is known about this plant in comparison with other species of Ilex, like Ilex cornuta, which played an important role in Chinese traditional medicine. in the treatment of dizziness and hypertension, or let's take an example of Ilex ficoidea or centrokinensis, which show potent anti-inflammatory and antioxidant activities. In the light of this information, it is worthwhile to investigate Ilex cassiana and uncover its treasure chest to find out its pharmacological importance. Among the 410 species under the genus index 2010, Ilex cassiana is a wholly endemic to the hills of Meghalaya, India. It's classified as critically endangered under the IUCN Red List of threatened species. The aerial, mainly the fruits, serves as fodder for wildfowls like squirrels and birds. And among the Khasi people, the bark and root decoction is used for the treatment of tuberculosis and severe cold. Even though it is known among the Mizo traditional healer, but the exact ailment to which the plant is used remains unknown. That is why we are drawing closer attention towards this plant, and new scientific finding regarding this plant will contribute greatly in knowing the plant better, as well as the need for its protection against exploitation. Next, please. All right, why this experiment? Novel experiments, as I mentioned earlier, that even if we did many literature survey, but not much experiment has been done on this plant. So all that we did is a novel experiment. So we performed preliminary as well as in vitro analysis for this plant. It is found to possess interesting results in its antioxidant, antimicrobial, antiparasitic, and anti-inflammatory activities. So we adopt the simple yet reliable method to screen the plant to extract the plant extract to find out the compound which are responsible for its medicinal values from the set of experiment that has been performed earlier. So yes, we are moving to the methodology. Here we are using HPTLC, which is a sophisticated chromatographic tool that produces fast and reliable results with superior separation efficiency. It includes validated methods for qualitative and quantitative analysis using the semi-automatic Linomet 5 applicator, and as well as TLC scanner, which is sensitive enough to detect 100 to 500 picogram of a compound. So result can be obtained within 3 to 20 minutes with 5 to 10 times better detection sensitivity. WinCat's software is used for all the instruments, to operate all the instruments. HPTLC fingerprint of herbal drug sample visually either confirms or rejects the plant identity. That is why HPTLC fingerprinting is really, really important. One of the fastest methods to identify a plant and its compound. In addition to ID, and HPTLC can also give some semi-quantitative information like intensity of bands. Quantitative evaluation of analytes can be achieved based on hyperspectral data, densitograms, or image profiles. Okay, next slide, please. All right. Sample is prepared by sequential extraction with three main solvents like petroleum ether, chloroform, and methanol using Soxhlet binding 72 on each solvent. The extract were dried and 50 mg was taken and 1 ml of methanol was added to it to get 50 mg per ml working concentration. From 6 microliter each was taken for sample application. HPTLC screening was performed for all three of them. Peak was not detected on both petroleum ether extract as well as chloroform extract. That is why we have chosen only the Ilex cassiana methanol extract, which is IKM. Next slide, please. All right. Extensive survey was performed and a lot of trials and errors has been done to find out the mobile phase. So finally, we chose this toluene ethyl acetate, formamide, and methanol in a ratio of 5.5:3:1:0.5, which is scanned, which we scanned at 386 nanometer, and this is the plate showing bands of multilayer and IK after scanning the plate with at 360 nanometer using UV visualizer. Okay, next slide, please. All right, this is the chromatogram, and the height and area of standard varies depending upon the amount of quercetin content. The chromatogram represents 300 nanogram of quercetin having 0.77 RF value upon scanning with TLC scanner. Next slide. All right, here 6 microliter of sample was taken from a working solution of 50 mg per ml concentration, and RF value of 0.77 was obtained, confirming the presence of quercetin. All right, next slide. Yeah, in this slide we see calibration curve that is plotted against the level of standard ranging between 100 to 300 nanogram, giving the calibration equation with correlation coefficient of 0.99, having a quantified value of 184.72 nanogram. Okay, the final now we'll come to the conclusion. Quercetin is a flavonoid found in many plants. Like we normally know that these plants like citrus, berries, leafy vegetables, spices, tea, and cocoa. It is a natural flavonoid found abundantly in vegetables and fruit. There is growing evidence suggesting that quercetin has therapeutic potential preventions and treatment of different diseases, including cardiovascular disease, cancer, and neurodegenerative disease. Quercetin has been known to exert antioxidant, anti-inflammatory, and anti-cancer activities in a number of cellular and animal models, as well as in human, through modulating the signaling pathway and gene expression involved in these processes. So the mobile phase gave us a good separation of the compound with RF value of 0.77. The presence of quercetin in Ilex khasiana might contribute greatly upon its medicinal value, namely its antioxidant and anti-inflammatory properties. All right, the final slide. All right, these are my references and my publication as well. And thank you so much.
Thank you very much, Charles, for your presentation. Now we will open the time for questions or comments from the expert members.
One small thing is there, although I am not of this field, but I just wanted to know, your studies shows that this plant, what you have studied, has got good anti-inflammatory and such things. So what do you think it can be used for some producing on a larger scale, on a industrial scale? Is it possible to use it, or it is being already used?
No, sir. As I mentioned, this plant is normally endemic to Meghalaya. Grown is found in Mizoram.
Okay.
We have to go for multiplication plus. We have to find out what exact compound is there. That is why I'm running HPLC fingerprinting to find out where skin is there.
So still you don't know the precise constituents which are of which can be used for pharmaceutical purposes. Still, it is to be explored. It is to be studied.
Yes, sir. For this purpose, we have to go for compound isolation, which is a tedious. So my next level of studies will be on that one.
Yeah. You need to extract whatever oil or whatever contents. Yeah.
Exactly,
yeah,
sir.
all right,
yes.
all right. Oh, it's okay. It's all right. So
yes.
At this point, I think maybe the last presentation may be there, but these presentations are quite quite good. But I would like to take leave from the organizers for organizing such a nice program during such a pandemic, using whatever resources. I think I should take leave now.
Thank you very much for your presence. We have just one last presentation, a poster presentation. If you can bear with us for just 5-6 minutes. We'll call our
All
next
right, okay,
presenter
okay.
and the last presentation for today, Elizabeth Vanelwati.
Yes, ma'am.
Are you able to share your slide, or we'll do it from here? It may be
Ma'am, if you
faster
can do
from
it from
here.
there.
Okay, okay. All right, are you able to see your slide?
Yes, ma'am.
All right, please start.
Okay. Greetings, everyone. My name is Elizabeth Valerwati Ngamlay. I'm a research scholar from the Department of Botany, Mizoram University. Today I'm presenting a poster on the topic in vitro antioxidant activity and preliminary phytochemical screening of aqueous leaf extract of Hedyotis scandens. So, Hedyotis scandens, it is a perennial climbing plant, and in Mizoram we call it Kilnamtur or Lichen Trebur. It has been used traditionally for as medicine for the treatment of stomach pain, ulcers, kidney problem, etc. And my main objective here in this study is to estimate the phytochemical components and the free radical scavenging property of the aqueous leaf extract of H. scandens. Okay, next slide, please. Okay. Okay, here that's my methodology. First, I collected and prepared the plant extract. Then after that, I studied the qualitative phytochemical screening using standard protocol. After that, I determined the total phenolic content, and then the total flavonoid content, and lastly, the in vitro antioxidant activity using the DPPH, radical scavenging activity. The first graph is the standard curve for total phenolic content using gallic acid as standard, and the second graph is the standard curve for the total flavonoid content using quercetin as standard. And the third figure is showing the DPPH activity of aqueous leaf extract of H. scandens against standard, and the standard used here is BHT. On my results, the analysis of the plant extract revealed the presence of compounds such as alkaloid, carbohydrates including reducing sugar, flavonoids and saponins, whereas some compounds like phytosterols, glycosides, tannin and protein were found to be absent. The total phenolic content was found to be 2 mg per gram gallic acid equivalent, and the total flavonoid content was calculated to be 6 mg per gram of quercetin equivalent. The Hedyotis scandens, the aqueous leaf extract of Hedyotis scandens, it has a radical scavenging activity by inhibiting DPPH with IC50 value of 6 microgram per ml in comparison to the standard BHT with IC50 value of 2 microgram per ml. Okay, and discussion. This study, it shows that the H. scandens may have potential, it has a potential antioxidant activity and can be further used in future for pharmacognostic use. Thank you.
Thank you very much, Elizabeth. Are there any questions for Elizabeth from the expert members? All right, if not, so we have come to the concluding part of our technical session for today. I will ask the expert members, any of the expert members who would like to give a concluding remark. The floor is yours.
Yeah. Yeah. So we had altogether around 9 presentations, maybe 2 from the life
From the time
sciences
you joined.
and 7
maybe.
from our side. Yeah.
Yes.
Yeah, and wide variety of topics, yeah, have been covered. So we are very pleased. I am pleased to be participating in this Congress, and I find that all the presenters were quite serious and they have given quite good presentation. Although the general comment is that I expect that all of them, whatever title is there, they should restrict their presentation to the title so that something good, concrete should come out for the benefit of the scientists as well as for the benefit of the societies where we live in. So with these words, I would like to convey my thanks to all the presenters and to the organizing team to have organized such a nice event. And really, it should lead into some publication so that it becomes more memorable and the results of some of the presentations can further up to get some data which may have some societal benefits. Thank you. Thank you very much.
Thank you very much, sir.
Okay. Congrats the organizers for the accomplishment we have just seen, and it's indeed an honor for us to be sitting as an expert in this day-long session. Thank you very much for, and I'm particularly feeling apologetic, guilty for my network is very poor out here. I have resumed my session more than 20 times. It's yeah, I'm apologetic for that, and congrats for your accomplishment and organizing this program. Thank you, madam.
Thank you very much. So we will now, before we leave the session, there are some announcements for all the presenters and the participants. The certificates will be sent to you through the email ID that you have provided. We'll be sending it to you in the next few days. And at 4 p.m., the closing function for the Mizoram Science Congress 2020 online will be happening, will be streamed online through YouTube. So if you are, we would be very happy if you could kindly join us and watch us on YouTube in the channel, Mystic Mizoram channel. And I thank all the expert members and our keynote speaker this morning who made your precious, in spite of your precious and busy schedule, to sit with us the whole day for this very, very long session and giving all the comments and suggestions to all the presenters. And I thank all the presenters for taking their time to participate and in this Mizoram Science Congress 2020. I wish you all the best in the future. Stay safe, and I hope to see all of you again participating in the next Mizoram Science Congress. Thank you very much. Bye-bye.